arthritis or rheumatism or whatever it is. So, our dialogue that is our discussion with Stina. There are many areas where this medium and excited and well-informed people like you can connect the world. Stina had a question. It is also a question asked in France. Beatrice Beatrice Deschamps എന്ന് പറഞ്ഞൊരു ആളുണ്ട്. ബിയട്രിസ് ഞങ്ങളുടെ കൂടെ ഉണ്ടായിരുന്ന ഒരു പ്രോഗ്രാമിൽ, പ്രസാദ്ജി ഉണ്ടായിരുന്നു ഞങ്ങളുടെ കൂടെ. ആ പ്രോഗ്രാമിൽ ബിയട്രിസിനെ ഫ്രാൻസിലുള്ള ചെടികളുടെ പ്രിൻസിപ്പിൾസ്, ബിയട്രിസ് എന്താ വെച്ചാൽ ഷീ ഹാസ് എ PhD in finance. അവിടെ ഫ്രാൻസിൽ PhD ഫൈനാൻസ് ഒക്കെ ചെയ്തിട്ട് she said, all this is nonsense. I want to study Ayurveda. And she came to study Ayurveda in Jamnagar. 6 varsham padichu, and she is so analytical. And she has that analytical mind. അപ്പോൾ ഇത് മാതിരി പല പല ആളുകളുണ്ട് പല പല ദേശങ്ങളിൽ. അപ്പോൾ അവർ നമ്മുടെ കൂടെ വരും. പക്ഷെ അവരുടെ ചിലപ്പോൾ അവർക്ക് ആ ഇൻട്രസ്റ്റ് കാണത്തില്ല തത്വങ്ങളും ശാസ്ത്രം പഠിക്കാനും. അവർക്ക് they don't have the skill also, Sanskrit and text. But I think we can develop a language independent representation of this science. My thinking, Murliji, namukku language illade we can still convey these principles. Yes, language, the texts are written in Sanskritam or other languages, commentaries are written, but we can enable a dialogue even without those principles. And for me, all of Ayurvedic principles are mathematical, very precise, combinatorial principles. If we start from Vyakt to Avyakt, we will see the progression leading to Mahagunas in the Avyakt to Mahagunas coming into the Vyakt. This is in my mind, this is what I see. Panchabhutangal, how do you generate the logic of the Mahabhutas? From there, how do we generate the logic of doshas? The bhutas are you can look at them as very precise entities. How to get those interaction between the bhutas to give you the doshas. Doshangal, from there, how to make life? This is a big question. For us here, the question is, why does life happen? This is a very important question. What is the reason for life? There is no reason why there should be life. The 5 bhutas, as we understand, they can stay just like that. They don't need to form anything else. But something is there that makes life come about. Once you make the life, you have different kinds of life. Adile ee pala jeevigal vannale adile oru jeevi is called Manushya, that can ask this question, who am I? So that philosophy is very important. From that question comes all this knowledge and the darshanas and understanding. So we have to provide everybody that initial foundation, which you people have learned as children, because you learned it from your father or your mother or your grandparents or your uncles or aunts or something. In our culture, it is easy to understand these things, but for a lot of people, it's difficult. That worldview is missing. So, if we give people a little bit of that appreciation, then we can start to open up Ayurveda without a language. There is a combinatorial thing about 5, 3 dosham. From there, we make life. From life, we make Manushya, and other pranis are there. The deep understanding of the concept of prana. What is prana? Prana, what is this prana? It is understood in different ways in different cultures. Namada yoga philosophy, they have pranas, and they say panchaprana, the 5 pranas. Our logic, Ayurveda, we have panchavayus. Prana aspect becomes vayus. People get confused. How come somebody is using panchaprana and you are using panchavayu, and some people are using subdosha of vat? or that's confusing there, you know. So that those principles we have to somehow clarify for ourselves so that we can present it to other people. To people who are alien to our culture. They don't understand. Most people don't understand. I particularly like Sankhya. Sankhya, Kapila 68 sutras, you know, couple of Sankhya sutras. That 68 sutras, if you understand many of this, the derivation is clear. And if we look at the aspects of the Sankhya sutras and the clarity of Vaisheshika Darshana, Vaisheshika says all dravyas are only 9. About our precision, you know, how do they get this? And a lot of people who are used to modern scientific measurements, you know, for them everything has to be measured. They cannot realize that qualities can be so powerful also. And Namade Vaisheshika Darshanayil Povumbum, Dik and time, direction and time are also dravyas. You know? And that is so deep a logic. Very, very deep. And it has so many day to day, we experience it every day. I'm writing something, and if I'm writing on a piece of paper, I give you one example. It's a practical example. Pencil. അപ്പോൾ ഞാനൊരു ചെറിയൊരു പേപ്പറിൽ ഈ പെൻസിൽ വെച്ച് വല്ലതും തുടങ്ങിയാൽ, ഇപ്പോൾ ഒരു ചെറിയ പേപ്പറിൽ ഒരു പെൻസിൽ വെച്ച് ഈ പെൻസിൽ വെച്ച് തുടങ്ങിയാൽ, I cannot continue this work without that pencil. That I'm stuck with this. I have to use the same pencil. ഞാൻ ഇവിടെ 3-4 പെൻസിൽ വെച്ചിട്ടുണ്ട്. എന്ന് വെച്ചാൽ പല പല ഈ ലെഡിന്റെ ഈ ഡെൻസിറ്റി, ഒരു ഹാർഡ് ബ്ലാക്കോ 2B വല്ലോ, there is different densities. അപ്പോൾ ആ ഒരു സെൻസേഷൻ, ഈ പെൻസിലിന്റെ ഈ ലെഡിന്റെ സെൻസേഷന് ഈ പേപ്പറിന്റെ അകത്തോട്ട് പിടിച്ചാൽ പിന്നെ I cannot continue this writing without that same pencil. So that is a quality, you know. And the other thing, ഞാനിപ്പോ ഈ മുറിയിൽ ഇവിടെ ഇരുന്നിട്ട് ലൈറ്റ് ഈ സൈഡിൽ നിന്നാണ് വരുന്നത്, ലെഫ്റ്റിൽ നിന്നാണ് വരുന്നത്. അപ്പൊ ഞാൻ ഈ എഴുതിയ കാര്യം ഇതിൽ കുറച്ചുകൂടെ ആഡ് ചെയ്യണമെങ്കിൽ ഞാൻ വേറൊരു മുറിയിൽ പോയിരുന്നാൽ നടക്കത്തില്ല. എന്ന് വെച്ചാൽ there is already a strange connection between light, which is a direction, which is a position, with what I am doing here, my function. You know, I'm carrying out a neuromuscular function of writing something. These sensitivities were known by these ancient people, so clear. And they could, if I have to write one shloka about this, I don't know where to begin. I don't know how to express it. But this is given in Vaisheshika Sutras. It is so amazing. It is all there, written in the sutra, in sutra form. That means so few words, and it is so precise. So this is the this is where I feel our logic and the clarity that is there is so deep and so sensitive and so perceptive about. And that is the beauty of your science. And your science has all that within it, you know. Nyaya, how to understand, how to understand the logical building up of aspects of this science. So my fascination is to see how to take this into the world of modern biology. പിന്നെ അവിടെ നിന്ന് മോഡേൺ ബയോളജിയിലുള്ള പുതിയ ടൂളുകൾ അതെങ്ങനെയാണ് നമ്മുടെ ആയുർവേദത്തിന്റെ അങ്ങോട്ട് എങ്ങനെയാണ്, how can we make that connection? നമ്മളിപ്പോൾ I'll give you one example. This is a very simple example that you will be able to clarify even more eloquently. Is the case of Basti, one of the karmas that you have. Basti is used routinely. You have a logic for using Bastis. And Basti is a very important procedure that you use for managing so many Vata-related conditions. So, ഈ Basti നടത്തുന്ന സമയത്തിൽ you have some technicalities. What is the dravya you use, etc., etc. And you have a logic for it, So something is, you have a body of knowledge, you have a technique, you use it, and it relieves some problems. Now, people who don't know any of these darshanas and this experience that you have, and the collection, the body of knowledge that is used for building the logic of Basti, they are completely confused. For example, Europe, I'll give you one example. Back pain is a big problem. And the reasons are many. The reasons could be improper bowel movement, delayed lower, the colonic transit time is very low. So, the gastroenterologist can tell you about transit time. They'll say, if you eat food, it takes so much time to go from this to this to this, which you also understand. You have a language for it. You will say it takes so many nimisha to go from here to here and from there to the next part, etc. And then it eliminates. So when the transit time is compromised, then people develop all kinds of complications. Now, for you, in your language, you understand this in terms of apanavayu, I guess. I think that is your understanding. Or there may be some other principles, but in a simple way, you would say there is something called apanavayu, and the intestine or part of the intestine is the home for vata, and we can manage it using this treatment. And you have a logic there. When we go into modern biology and you say, how does this work? Can we measure transit time? Yes, that is easy. Can we measure nerve conductance? Now, colon, the vertebral column, that where there is well-established nerve connections. So you can start to measure those aspects. And then more excitingly, you will say many other attributes which are connected to bala or some aspects like that. The modern biology says, how are we going to measure all this? Well, there was one study conducted in Bombay where they measured many cytokines. So neurology is easy to measure cytokines also. And what did they find? That after Basti, almost for 138 days after you have completed a Basti protocol, the pro-inflammatory cytokine called interleukin-6, the level keeps dropping and dropping and dropping. This is so amazing. On one side, you have a procedure that is so common for you people. Basti is a routine procedure you would use routinely for many conditions. On the other side, there are people who don't even know what is Basti. And for some people who know, they can make this bridge between Basti, nerve conductance, feeling light. You use terms like you will feel light, and you can make connections with IL-6, which is a pro-inflammatory cytokine.
Excuse me, Madanji.
Oh.
Shall we get into the session now?
Please, please, please. I have taken.
Rajmohan is eagerly waiting for that.
Thank you.
Okay. Actually, I was also listening to Madan sir. Karthik, please have a formal.
Introduction by Karthik, please. Then we will get into the research topics.
Thank you.
Namaste, everyone. Actually, Om Prakash ji had asked me, should we start streaming it after the formal introduction? So I told there is no need of that. We shall directly start streaming because we have already got into the point. We have already come to the point. And even in this short duration, the points and the impact of the points conveyed by sir was very large, especially regarding the qualities, impact of darshanas, the necessity of understanding darshanas for a proper deciphering of whatever the wisdom has been mentioned in Ayurveda. So they are great messages to the students as well as practitioners in Ayurveda. So we have already started streaming. So coming to a formal introduction. Sir, I hope I shall continue in Malayalam, sir.
Please, please, please.
For the introduction.
Malayalam, I understand everything സംസാരിക്കാൻ മാത്രം കുറച്ചൊരു ഡിഫിക്കൽട്ടിയാണ്. പക്ഷേ പ്ലീസ് കണ്ടിന്യൂ ഇൻ മലയാളം. ഐ ക്യാൻ ഓൾസോ ടേക്ക് ക്വസ്റ്റ്യൻസ് എവറിത്തിങ് ഇൻ മലയാളം. ഇറ്റ്സ് ഓൾറൈറ്റ്. ഇടയ്ക്കിടയ്ക്ക് ഞാൻ മലയാളത്തിൽ കുറച്ച് I will also use a little bit of Malayalam.
Sure, as per your convenience. എല്ലാവർക്കും നമസ്കാരം. ആയുർവേദത്തിലെ ഗവേഷണ സാധ്യതകൾ അതിനെക്കുറിച്ച് ഉള്ള PG വിദ്യാർത്ഥികൾ, വൈദ്യന്മാർ, അധ്യാപകർ ഇവർക്കിടയിലുള്ള ഒരു ബോധവൽക്കരണത്തെ ഉദ്ദേശിച്ച് നടത്തുന്ന ഈ ഒരു പരമ്പരയിൽ 3ാമത്തെ ക്ലാസ് ആണിത്. അപ്പോ കഴിഞ്ഞ ക്ലാസ്സിൽ ഡോക്ടർ KV രാധാകൃഷ്ണൻ സാറുമായിട്ടുള്ള സെഷൻ ആയിരുന്നു. അത് ദ്രവ്യ കേന്ദ്രീകൃതമായ പഠനം, ഫൈറ്റോ കെമിസ്ട്രി അതുപോലത്തെ വിഷയങ്ങളെ കേന്ദ്രീകരിച്ചായിരുന്നു ചർച്ച. അപ്പോ അതിൽ മദൻ സാറും പങ്കെടുത്തിരുന്നു. അപ്പോ അന്ന് അദ്ദേഹം സൂചിപ്പിച്ച ഒരു ആശയം ഉണ്ടായിരുന്നു. We must have a dialogue with the contemporary sciences in order to update our Shastra. So I think the main theme of today's conversation will be mastering this art of dialogue or a transdisciplinary dialogue. So and of course with a special focus on themes like molecular biology, genomics, cellular biology, systems biology, etc. So അതിന് ഇന്ന് നമ്മളുടെ ക്ഷണം സ്വീകരിച്ച് ഈ ഒരു ചെറിയ ഈ ഷോർട്ട് നോട്ടീസിലും നമ്മുടെ ക്ഷണം സ്വീകരിച്ച് ഇവിടെ എത്തിയിട്ടുള്ളത് ഡോക്ടർ മദൻ തങ്കവേലു സാറാണ്. സാറിന് പ്രത്യേകിച്ചൊരു ആമുഖത്തിന്റെ ആവശ്യമില്ല. He is very popular among the Ayurvedic community as well as the global scientific community. Even in that case, giving a short intro. So he's a world famous genome biologist. He has done his PhD in molecular genetics from Cambridge University. And also is having a fellowship in the field of oncology as well as molecular biology. And he has invented a technique called molecular copy counting technique. I think it is concerned with deciphering the differences or variances at the genome level or gene level on a cellular basis. I think he will elaborate on that in the session too. And also linker ligated hyper efficient whole genome amplification technique. And he is also a member of Mind Matter Unification Project, Cavendish Laboratory, Cambridge. And the way he is associated with Ayurveda, he is currently the research director of European Ayurveda Association. And even in India, he has been working tirelessly in association with organizations like Ayu, the Ayu Journal from Jamnagar, and Ayu Care Case Report Journal from All India Institute of Ayurveda. And he is also an associate with the Transdisciplinary University Bangalore, as he had mentioned in the beginning of the session. And with this short introduction, actually there are several accolades that are to be added, even though welcoming you on behalf of this meeting, sir. Namaste.
Thank you. Namaskaram. Thank you, Karthik. Happy to be here with everybody. Now, I like this format that we were streaming even before the formal introduction. And what you said is nice, that we touched on some very key points. Now I leave it to you to say where you want me to go.
Yes, sir. Yes, sir. And today's moderator, Dr. Rajmohan sir. Rajmohan സാറിന്റെ അദ്ദേഹത്തിന്റെ ഉത്സാഹം തന്നെയാണ് ഇതിനെ പ്രധാന കാരണം. അപ്പോ ഈ ഒരു ഇൻഫർമേഷൻ നമ്മൾ ആവുന്നത്രയും ആളുകളിലേക്ക് എത്തിച്ചിരുന്നു. നമുക്ക് 60ഓളം പേരിന് എത്തിയിട്ടുണ്ട്. എനിക്ക് തോന്നുന്നു ഇതിൽ പ്രധാനമായും ഈ സെല്ലുലാർ ലെവലിലുള്ള ഗവേഷണത്തെ കുറിച്ച് കൂടുതൽ അറിയാനായിട്ടും പ്രത്യേകിച്ച് ദ്രവ്യ പഠനം അതിലൊക്കെ കേന്ദ്രീകരിച്ചിട്ടുള്ള ആൾക്കാരാണ് ഇവിടെ എത്തിയിട്ടുള്ളത് എന്ന് തോന്നുന്നു. അപ്പോ ഏതായാലും രാജ്മോഹൻ സർ, അദ്ദേഹത്തിനെ കൂടുതൽ പരിചയപ്പെടുത്തേണ്ട ആവശ്യമില്ല. ഗവൺമെന്റ് ആയുർവേദ കോളേജ് തിരുവനന്തപുരം രസശാസ്ത്രം ഭേഷജകൽപ്പന വിഭാഗം അസോസിയേറ്റ് പ്രൊഫസർ ആണ്. അതുപോലെ കോവിഡിനെ സംബന്ധിച്ചുള്ള സ്റ്റേറ്റ് ആയുർവേദ കോവിഡ് റെസ്പോൺസ് സെല്ലിന്റെ സ്റ്റേറ്റ് ലെവൽ കോർഡിനേറ്റർ ആണ് അദ്ദേഹം. അപ്പൊ അദ്ദേഹത്തെ അനൗപചാരികമായി തന്നെ ഈ വേദിയിലേക്ക് സ്വാഗതം ചെയ്യുകയാണ്.
Thank you, Karthik. Madan sir, good evening, everyone.
Namaskaram.
Okay, good evening. So, shall we start?
Thank you, Karthik.
Okay.
Yes, sir. Sir, നമുക്ക് കൂടുതൽ നമുക്ക് വേണ്ടത് ഇന്നത്തെ ഒരു ഡിസ്കഷൻ, ഓഫ് കോഴ്സ് യു ഹാവ് എ വാസ്റ്റ് ഏരിയ ഓഫ് എക്സ്പെർട്ടൈസ് ഇൻ മോളിക്കുലർ ബയോളജി ആൻഡ് ആസ് വെൽ ആസ് ഇൻ സിസ്റ്റംസ് ബയോളജി. Of course, sir is also associating with Ayurveda for quite a long time. So ഇവിടെ PG സ്റ്റുഡൻസ് ആണ് കൂടുതലായിട്ട് ഇവിടെ എത്തിയിരിക്കുന്നത്. അപ്പോൾ എനിക്ക് തോന്നുന്നത് അവർക്ക് കുറച്ചും കൂടെ മെത്തഡോളജിയിലേക്ക് എത്താനും കുറച്ചും കൂടെ ഫീസിബിൾ ആയിട്ടുള്ള റിസർച്ച് സെലക്ട് ചെയ്യുവാനും അവരെടുത്തിരിക്കുന്ന റിസർച്ചുകളെ കുറച്ചും കൂടെ ഫ്രൂട്ട്ഫുൾ ആക്കി എങ്ങനെ എത്തിക്കാം എന്നുള്ളത് കൊണ്ടാണ് എന്ന ഒരു ആസ്പെക്ട് ആണ് നമ്മൾ കൂടുതലായിട്ട് ഇവിടെ കവർ ചെയ്യാനായിട്ട് ഉദ്ദേശിക്കുന്നത്. സോ സാർ അതൊന്നും മനസ്സിൽ വെച്ചതിന് ശേഷം അവരെ ആ ഒരു മെത്തഡോളജിയിലേക്കും അവരെ അങ്ങോട്ടൊന്ന് അട്രാക്ട് ചെയ്യുന്ന തരത്തില് സാറിന്റെ ഭാഗത്ത് നിന്ന് കമന്റ്സ് ഉണ്ടാകണമെന്ന് അഭ്യർത്ഥിക്കുകയാണ്. സോ ടു ബിഗിൻ വിത്ത്, ഡെഫിനറ്റ്ലി നമുക്ക് എല്ലാവർക്കും സാറിനെ അറിയാം. സാറ് എന്നെ എത്രത്തോളം മോട്ടിവേറ്റ് ചെയ്തിട്ടുണ്ടെന്നുള്ള ഒരു കാര്യം ഞാൻ മുമ്പ് കഴിഞ്ഞ ഇവിടെ പറഞ്ഞിരുന്നു. ബാംഗ്ലൂരിൽ എലഹങ്കയിൽ എനിക്ക് തോന്നുന്നു ഒരു രാത്രിയിൽ ഏകദേശം 1 മണിക്കൂർ മാത്രമാണ് നമ്മൾ ഉറങ്ങിയിരിക്കുന്നത്. ബാക്കി മുഴുവൻ സമയവും actually I was introduced into the fascinating world of molecular biology and how Ayurvedic principles can effectively be demonstrated within a cell, how it exactly merge, these principles exactly merge with some of the complex and intricate molecular mechanisms. How you can demonstrate that all those things actually Sir was exposing us towards that. We had been discussing about 3 beautiful projects. One project was how we can demonstrate the fundamental principles of Ayurveda more objectively. That was one of the project, like Panchamahabhutas and Tridosha theory, etc. And the second one was it was regarding diabetes, its molecular aspects and prameha. That was we were discussing. And the other one is the other major project in which Madan sir and I was involved was actually Rasayana, aging and cancer. That was the high impact project, 3 high impact projects. We have developed a pretty good project in that, but in these subjects, but unfortunately it didn't come out good. But probably in the coming years, definitely it will come again. It will become fruitful, I believe. Sir, to begin with, സാർ ഒരു മോളിക്കുലർ ബയോളജിസ്റ്റ് ആണ് എന്ന് ഒക്കെ നല്ലൊരു ഇൻട്രൊഡക്ഷൻ ഇവിടെ നമ്മളുടെ കാർത്തിക് പറഞ്ഞു. എനിക്ക് തോന്നുന്നത് പ്രോബ്ലി ദ സ്റ്റുഡൻസ് വിൽ ബി വെരി മച്ച് ഇൻട്രസ്റ്റഡ് ടു നോ ഹൗ യുവർ ഇൻട്രസ്റ്റ് ടേൺഡ് ടുവേഴ്സ് ആയുർവേദ ഓൾസോ. നൗ ഐ തിങ്ക് യു ആർ ദി one of the leading propounders of Ayurveda globally. സാർ ഒരു ആയുർവേദത്തിനെ വളരെ കാര്യമായിട്ട്, സാറിനെ പോലെ കേംബ്രിഡ്ജ് എന്ന് പറയുന്ന വളരെ ഫേമസ് ആയിട്ടുള്ള ഒരു യൂണിവേഴ്സിറ്റിയിൽ നിന്ന് വളരെ ഒരു റെപ്യൂട്ടഡ് ആയിട്ടുള്ള സയന്റിസ്റ്റ് ആയിരിക്കുന്ന അങ്ങേ പോലെ ഒരാൾ ആയുർവേദത്തെ കുറിച്ച് ഇങ്ങനെ സംസാരിക്കുന്നത് തീർച്ചയായിട്ടും ആയുർവേദത്തിന്റെ ഒരു അംബാസിഡർ എന്നുള്ള നിലയിൽ ആണ് സാറ് ഇന്ന് കാണപ്പെടുന്നത്. എന്താണ് സാറിനെ ആയുർവേദത്തിലേക്ക് തിരിയാനുണ്ടായ കാരണം? അതൊന്ന് പറയുന്നത് തീർച്ചയായിട്ടും ഇറ്റ് വുഡ് ബി വെരി motivating for the students also. Sir, please.
Thank you, Rajman. Janet, English lip. I will continue in English.
Please. Please, sir. Please,
so, sir. I wish I could continue in Karthik and Rajmohanji, Prasadji. Thank you so much, number 1, for having me here. Number 2, I wish I could talk in Malayalam because our students will feel more comfortable to that, but it doesn't matter because they can still ask questions in Malayalam. So we'll keep it open for Malayalam. I also see Dr. Selvan Sentamil has joined us from Bethesda in Maryland. You will remember Selvan because he was with us also in Elanka.
Selvan sir, welcome sir.
Selvan is here. Namaskar. Selvan, thank you.
Namaskaram. Thank you.
So nice to meet you, sir.
Selvan is here. So Selvan clearly understands Tamil, but he might not easily follow Malayalam, so we will keep it to. But Selvan, please excuse if we transfer a little bit into Malayalam also because it's like
no problem. I understand a little bit, no problem. Tamil and Malayalam has kind of some relationship, but I can understand. Thanks.
Thank you. So happy to be here. Now, question: how did I come into linking with Ayurveda? So. Historically, the family has a lot of Ayurveda connection. So on my mother's side, my mother's mothers on the maternal side. My mother's maternal uncle was Ayurveda and a modern medicine person. His name is Captain A.N. Panikar, and they come from near Kollam. That's their hometown, and the Panikar family comes from there, and they studied medicine. He studied medicine. He was working here in Edinburgh, came to study and then was working in Edinburgh. And 1929, the government, the then British government in Sri Lanka, they wanted to establish a Deshiya Chikitsa school, because Sri Lanka has a tradition in Ayurveda that goes back to the time of Ashoka, you know. Ashoka's daughter and son, the Buddhists, they were sent to Sri Lanka to take Buddhism to Sri Lanka. So that was an old connection. And Deshiya Chikitsa was going on in Sri Lanka from that time. So Captain Panikar was invited to come and establish the school, Deshiya Chikitsa school in Sri Lanka. So that was 1929. And then, so the family has a lot of those connections. But I was born much later, you know, 30 years after that event in Sri Lanka. So that's when I was born in Trivandrum. I was born in Trivandrum in the newly established Sri Chitra Hospital. I was one of the first babies to be born in Sri Chitra Hospital in Trivandrum. 1959 was when they established the pediatrics department and that hospital. The Maharaja established that hospital with the help, because of some family reasons. They lost one child to rheumatoid diseases, and that's how they established this hospital. I was one of the first babies to be born there. And I studied in Trivandrum till in Srigariam, at the first in Holy Angels Convent, which is near where you people are, and also then in Srigariam Lila School. And then in 1972, my father went to work for the WHO in New Delhi, and we all went to New Delhi. I finished my schooling there. And then, to cut a long story short, in 1982, I came here to Cambridge. And I've since worked with DNA, always with DNA. I like DNA, and that's the reason why I came here. About 1999, I got the chance to come to work in the lab where DNA structure was discovered, the Watson Crick lab. And they were working at that time to understood, sorry, Murliji has just connected SAT Hospital, Outam Tirunal Hospital, not Sri Chitra. So in 99, we started working in this laboratory, the DNA laboratory, fine analysis of DNA. That means DNA, can you analyze single molecules of DNA? So if you look at every single cell in the human body, it has DNA contributions from the father and the mother, 2 chromosomes, one from the father, one from the mother, and the mitochondrial DNA that comes only from the mother. And when we start analyzing DNA at such detail, because that was part of the mandate to high sensitivity analysis DNA, you start to see DNA changes that you expect only in cancer or other DNA diseases. But these are found at very low frequency in our normal body. So it's happening there. At that level of single cells, you see DNA changes that appear traditionally connected with DNA diseases. So this was the first question, saying, how come our body has these mutations, DNA mutations, but we don't see the disease? There is a conflict here between DNA theory and our understanding of human biology when you see these observations. And you then say, will these conflicts go away? So maybe it is an experimental error that these mutations that you're seeing. Can you refine that experiment and get rid of these errors? And you try refining the experiments, but these errors don't go away. These are normal errors in DNA, which means your body, this sharir, has within it an opportunity to have so many changes hidden away, but the body is still working. We don't see the disease. The body is still working. There is no physiological consequence of those small changes. That means there are a lot of things happening at a lower level of resolution. And is this, do we have to live with it, or is it an error in the theory? Some things point to incompleteness in our theory of DNA and genomic theory especially. And then automatically we say, well, if this is incomplete, is there a better theory? Is there a more complete theory that can contain this, and where these small changes become irrelevant? And then I look around at that point and I say, there is Ayurveda, which is a complete theory of life. It explains everything in life. It does mention things about contributions from the father and the mother to make the prakriti, etc. But the emphasis is not so much. The emphasis is more on another language of how life works. And then I start learning about Tridosha. I learn about prakriti. I then learn about Panchabhutas and how you construct this theory. And then I go into the darshanas and I see how this logic can give rise to a body of knowledge that is used for understanding health, that is used for understanding promoting health, understanding how to prevent disease, understands how to cure disease, and all the different ways in which you handle these things. A body of knowledge that is very ancient and internally consistent. There are no errors in this body of knowledge. So I came to it through seeing an error in the DNA theory. DNA theory has an error, and to contain that error, I look for other theories, and there comes Ayurveda. So I am still learning about Ayurveda. I follow the Samhitas, Charaka Samhita, Shushrut, Ashtanga, and I haven't reached those Lagutriyas yet, Madhav Nidan. These are things that I would like to, but as I'm not a clinician, I feel I don't want to go too much into this area. I want to understand more of the theory of it and how that theory can be used to refine the theory of biology itself. So more recently, I am looking at this interface between Ayurveda and yoga to understand the concept of prana, for instance. Ayurveda agrees on the theory of prana, but you don't dwell too much on the aspects of prana. While you go into yoga, you will see the aspect of prana understood much better. There is the aspect of chakras, there is the aspect of nadis, there are aspects of understanding the 72,000 nadis that are flowing, where the energy flows, and the concept, the Panchakosha theory of yoga, which clearly Ayurveda doesn't go into it because you are only dealing with the Annamaya Kosha. But yoga takes you into a completely different level. And that level connects deeply into the deeper principles that are understood in the Vedas, that are understood in the Upanishads. And it takes me into all of those aspects. So I'm still a learner, I'm still learning, but I see the patterns. And I'm hoping that students who are coming into Ayurveda will come to it with that level of wish and desire, that they are studying something, and that study is a body of knowledge that is so complete and so extensive. And it takes you, if necessary, into the other areas of yoga, yoga darshana, understanding simple things like Swastavrit that you have, and also yoga connects in a slightly higher level of understanding. So this is my journey into Ayurveda and the Ayush systems. I also connect here in Cambridge with the Mind Matter Unification Project that is directed by Professor Brian Josephson. Brian is a very special man for us. I call him a rishi. If there is a modern day rishi, then it will take the form of people like Brian Josephson. We had another person here, Sir Aaron Klug, who was also a modern day rishi. Their understanding, their ability to see the detail and the big, the sthula and the sukshma. They did it. Although they didn't understand Sanskrit, they operated at the level. Sidney Brenner is another name here. He could see the finest of the finest and the largest of the largest. Now you are fortunate that all of you are brought up in that parampara to understand Sukshma and Sthula. And you have a lot of techniques that you use to understand at these levels. You have amazing techniques like Panchakarma. This is very special. It's such a special technique that has the ability to open up aspects of physiology that is not available to modern physiology. You have interesting concept. I talked earlier about the Panchavayus, how you understand the movement of energy in the body, and how you nicely connect the concept of energy with physiology. And you practice this. It is not just theory. You get patients, you can treat them, you can give them certain dravyas. You have certain ways in which you can manage and manipulate the physiology of the human body. So I am excited for all of these reasons that you have so many things that you are taught, you can improvise on, and they're thousands of years old. And this body of information and this body of techniques is what you are using every day. And I feel ashamed that there are people in the world who don't know about this. And so that is what I do. I try and see how to connect these two systems.
Okay, sir. Okay.
Thank you.
Thank you so much, sir. That was pretty well explained how you came to this Ayurvedic side. And of course, you are also a part of a great parampara that is continuing with this thinking and all. Thank you so much for being with us, sir. I have the next question to you is, actually, as a molecular biologist, how you perceive the principles of Ayurveda in the systems biology perspective or cytogenetic aspects? Is there any conflict? Because most of the time when it comes to Ayurvedic principles, some of the scientists are telling that the objectivity of some of these principles are very less, and you may not be able to demonstrate it. I don't know how we can demonstrate life. I don't know how we can demonstrate even time, because we can only perceive its existence only. So you are a person who is very, very much working on this DNA aspects and the minute aspects of life. Is there any conflicts you are facing with these principles, or whether you are finding any difficulty in corroborating and correlating these principles inside this with cytogenetic aspects and all, sir? How do you feel?
Thank you, Rajmohan. Another very important question. Early this morning, I was talking about some of this with Selvan. We were having a very heated debate about this stuff. So
Okay.
Happy Selvan.
Probably Selvan sir also can
join
contribute his thoughts in this.
Now, your question is very important, and it's been told a long time ago by the person, Erwin Shergaff is a very important person in the history of DNA. He was a German scientist who went to New York and worked in New York for many years. And Shergaff gave an early rule called Shergaff's rules. And Shergaff's rules, that was a time when we knew how to isolate DNA, but we didn't know what was this thing called DNA. And Shergaff's rule was analysis of DNA. Okay, so DNA, when Shergaff he was a chemist, he could analyze DNA and he could see the two bases, purines and pyrimidines, okay? Now, Shergaff's rule was that the ratio of purine and pyrimidine is always very tight. So I'm taking you a little bit into the history of DNA analysis, and Shergaff's rule says there is always a tight ratio between purines and pyrimidines. Now, this was 1940s. The structure of DNA, which tells us about the double helix, gives us the detailed explanation of the ratio of purines and pyrimidines. It says A's will always pair with T's, and the G's will always pair with C's. And therefore, there is a tight, if I use a term from your language, I would say a sambandh, a samavaya sambandh between purines and pyrimidines. So here is the first expression of your language in terms of DNA, samavaya sambandh. You cannot separate these two. Purine, pyrimidine has the samavaya sambandh. Now, this is one example of how we can see the conflict and how we can resolve the conflict. Objectivity in Ayurveda. So the question is, all of Ayurveda is based on qualitative logic. You use qualities. Vaisheshika teaches you that, qualities. A quality is something that has an opposite. Something is warm because there is something colder than it or warmer than it, the opposites. And you use the principles of the 20 Vimshati gunas. You have the principle, you know, a guna is known because there is an opposite for it. Now, if a scientist is not familiar with the guna concept, and if they come to and look at Ayurveda or even Siddha medicine, and they come and look at the system and say, what are you people doing here? Why aren't you measuring something? And they don't understand that you are already measuring based on gunas. You have the two opposites. Every dravya can be reduced down to gunas. You see, you could take a dravya, the dravya has the guna aspect, and every guna has its panchabhutic aspects to it. So you have a way in which you manipulate things, you see, which is just like what you would do in quantitative sciences, where you have to measure something. But you have qualities that are equally powerful. So people then say, how will you give us objective evidence for panchakarma? How do you know this works? You have lakshanas for something. How do you know why, when you do snehapan, the first step, the purva karma, how do you know it is when to stop? You're using some quality, you're using lakshanas. But if there is this group of people who don't understand those intricacies, they will not get the game, they will not understand. So the challenge is that we are not explaining. I feel there are times we are not explaining things to people, saying this is panchakarma, this is the meaning of a karma. And this is why we do it. We start off with this karma. Or if you're doing any, I mentioned earlier about basti, you have a logic for why you do basti. When you do basti treatment, you have lakshanas. You are not doing it at random. You are measuring something also. But your measurement is based on a quality, a lakshana. And you have different lakshanas that are open to your five indriyas. So it might be whatever, it might be touch, it might be sight, it might be sound, it might be something. You are also have, if you go into your parikshas, you have examinations. You have roga pariksha, rogi pariksha. If somebody doesn't understand the word roga and rogi, then we have lost the game completely. You know, their complaints are valid, but we have to explain to them that within roga pariksha, rogi pariksha, we have Ashtasthana pariksha or Dashavida pariksha, and these are examinations. Yes, you can then use a stethoscope to listen to something, which is only an added instrument to amplify a signal that is already part of our analysis. You have Nadi pariksha. Nadi pariksha is so sophisticated. You have not only Vata, Pitta, Kapha on the three fingers, but you have 7 levels of nadis. You can do pariksha at 7 levels. And when you are even more talented, you don't even need to feel the nadis. You can see so many things on the skin texture, the hair texture, so many things you have, so many things. So the point here is, it might seem like there is a conflict between the qualitative logic and the people who are wanting objective assessment. But if they care to understand, what you mean by a lakshana, and if you are confident to explain to them, this is the logic of a lakshana. When we do snehapana, this is what happens in the body. And this is the reason why we do the purva karma. So your language must be taken to the other side. And you shouldn't be needed to explain all of what you're doing in that other language. There isn't a need for it. This is my personal take, but I feel there is something there from the people who are calling for objective assessment. So somebody has studied modern medicine. All they have learned to do with the stethoscope is to listen for murmurs or listen to the, but your Nadi Pariksha gives you so many other details. Yes, not everybody might be a good Nadi Vigyan. That is a different issue. But There are people who can read the nadi and tell you the function of your intestine. You know, they can read something there that is based on a qualitative analysis. So again, I remind you that whether you have the skill, whether all the students have that skill is a different matter. But your logic based on the darshanas and the tattvas builds a structure that enables anybody to do that kind of investigation. Is it? I don't hope I've conveyed my, my
it would
sentiment. So the question then is for the teacher to ask, do all my students understand it? And the question then to the teacher is, do you understand this also?
I have another question to add to this, because if a student really need to demonstrate some of these doshic aspects or some of our principles in a cytogenetic level or a cellular level, is it possible, or what method probably would suggest for such students, or is it feasible such studies? And just because there were some attempts, there were some biochemical correlations and some genomic correlations with the dosha prakriti, and some aspirin particular specific enzymes. Some demonstrations were there. Do you think that is it feasible, or what method probably you would suggest if a student really want to get into these kinds of researches?
No, thank you, Rajmohan. So let me add to this question. You have to understand the human body in its complexity. So that is the first step. The human body is an extremely complex structure. And that complexity is built at many, many levels. You have to appreciate that it's like a helix, you see. This complexity can be understood like a helix, and a similar stage at each level of the helix. So if I take a simple concept of Panchabhut Siddhant, to start from your side, I take simple Panchabhut Siddhant, and you take it to the different dhatus. You already say that different dhatus have predominance of different bhutas. If you go to Asti Dhatu, you already say it is predominantly Vayu, because there is something there in terms of the structure that is understood by you people. You know it, and your Rachana Sharir people have taught you all these things, and you understand these things. You then are saying, can we see Panchabhut aspects at another level in the human body? So you are taking Tridosha, Panchabhut Siddhant, can we see it at another level? Panchabhut, of course, you can see it at any level. Wherever you go in, you will see Panchabhut, Panchabhut. That is all that is there anywhere in the human body. Now, the question is, can you see Tridoshas at different levels? If you go into the modern medicine man might say, well, I have a pancreas here. Can you tell me where is Tridosh here in the pancreas? So this is a challenge. And that challenge is easy to resolve, because the three doshas, when your logic says when the three doshas are in balance, you will not see any problems. The tissue is working normally. But if some aspect of the three doshas are compromised, you will start to see problems. And let me give you one example. If you say, I want to understand what is kapha dosha, you cannot analyze kapha in isolation. Kapha has to be understood in terms of some other qualities, because kapha is defined not as kapha in isolation. Kapha is defined in terms of the other aspects. You have to understand kapha in terms of Agni. So whenever you say kapha, you are saying, you're making a relational step to other attributes. If you say kapha, increased kapha, are you implying anything about Agni in that statement? I feel you are. Whenever you talk about Vat, are you implying something pointing towards something else? Yes, you are. So we cannot understand those principles in isolation, but you have to understand it as connected to physiology, as connected to something else. So go back to the pancreas example. somebody says, I study the pancreas. Can you tell me something about where the doshas are in the pancreas? You can start off with a very simple example of the endocrine and the exocrine function of the pancreas. And you say, well, the image that I always refer to is one of the images in my presentation that's on the website, and it's about the exocrine function of the pancreas. You know, it produces amylases and all these kind of things that are pushed out into the pancreatic duct. If you look within that, you will find a layer of cells, a very fine layer of epithelial cells at the lining of every duct, okay? And if there is a malfunction in the three dosha aspect of that cell, the epithelial lining, one layer of epithelial cells, you will compromise the function of the pancreas. Now, all of a sudden, you see a srotodushti here. Two types of srotodushti. You see a dushti in the ductal activity. You see a dushti in the transfer of material from the inside of the pancreas to the duct. If you give me an opportunity to share a screen, Rajmohan, then I will pull up one image that you have seen before. Maybe the students would like to see this.
Yeah,
and it's it's a presentation I made in Kochi many years ago. And within that is, can you see my screen, Rajmohan?
No, sir.
Not
Karthik,
yet?
I hope sir is the co-host now. Right?
Karthik has given me, yes. Karthik has given me. Thank you, Karthik. I have. Okay, so this is I'll put this in our chat. I'll put this reference in our chat, and I will take you to, it's a presentation has many elements there, but I will take you to one slide in here, which is somewhere for, okay, here it is. This is a section of a pancreas, okay? Karthik, can you see my screen?
Yes, sir.
Yes.
Okay, so here is what I mean by a shrota. So for me, I'm taking the principle of shrotas into the pancreas to address the question, can you show me where is tridosha in the pancreas? So here is a transverse section across the pancreas. And here, these cells are lining the pancreatic duct, okay? This is the duct. So here is a section, and this is the duct of the pancreas. So things can come from inside the pancreas through these cells into this channel. It's a small shrota in the pancreas. What has happened here in these cells? You see this cell, if you see my pointer, one cell is this big. It has become, from a cuboid cell, it has become an elongated cell with a blue material inside. Now, this blue material is something that is blocking the movement of things from the inside of the pancreas to the duct, to the shrota. So, in fact, the cell itself it's full of shrotas, because that's how things come in from the inside of the pancreas to the channel. Something has blocked here. Now, for me, just this cell and this compromised activity is an expression of imbalance in tridosha. There is an imbalance here that makes this, that blocks the shrota of that epithelial cell. And this can be observed using a simple stain. In this case, it's just a blue stain that is used for staining glycoproteins. So the glycoprotein there is compromising the function of the pancreas by compromising the function of the epithelial cells. Now, every time you compromise a function, it is an imbalance in the three doshas. So here is a simple presentation of where you are bringing the three dosha concept down to one organ of the human body, down to one function, which is the exocrine function of the pancreas. And here is evidence for it. Now, if we go to the next slide, you will see what I mean by dosha imbalance. The only reason why this imbalance happens is because the doshas are disturbed. And what will you do when doshas are disturbed? You have two chikitsas. You can do shodhana or shamana. So we have to jump between those two areas. You're doing shodhana or shamana. And for me, what is happening here can be cleared out using your panchakarma process. There is a blockage here. I want to clear out the shrotas, and the shrota blockage is at many levels. Pancreas is not functioning. The pancreas is not functioning because the epithelial cells are not functioning. And because the epithelial cells are not functioning, the reason why the epithelial cells are not functioning is most likely a compromised Agni somewhere. Or there is some problem. There is Agni is compromised. If an Agni is compromised, Ama is created, which is your logic. And this is how I see Ama. This is Ama. And what do? You have to do Agni Deepan, Ampachan, and you have to clear the Shrotas. So first start off with Agni Deepana, Ampachana, whatever dravyas you give, and then you clear the Shrotas with whatever karma that you use. So here is at the level. So Rajmohan, in reply to your question, this is an explanation of Tridosh Siddhant taken into organ function at the level of single cells. I haven't lost any of those concepts. I'm still using Agni. Agni is compromised in an individual if pancreatic function is compromised. Agni is compromised in the pancreas if some function is compromised. Agni is compromised in the epithelial cell lining of the duct because of some problem. So when you do a karma, Ampachan or Agni Deepan, you're trying to reconnect and refresh all these problems. And the same could be done for the liver. The same could be done for your Mahashrota. The same can be done for your lung. The same can be done for your Majadatu. So the question is, at what scale are you doing this examination? And whether your karmas are going to work at all those levels. My feeling is that the concept of karma is like every other concept of Agni, they are scale independent. It doesn't matter. You can understand Tridosha at the level of the body. You can understand the same Tridosha at the level of individual organs. You understand Tridosha imbalance in dhatus. So you have Dhatvagni, and you say, okay, this Dhatvagni has to be improved, so we give this Aushadhi. You can take the same language. I put this in the context of pancreas. And I've said, okay, pancreas, the man who studies pancreas says there are two functions, endocrine and exocrine functions. I can use the same thing. I can use the same logic for the beta cells and the alpha cells of the islets of Langerhans. The same logic will work there. Agni is compromised, the cell doesn't function, so function is compromised. And then it scales out to the whole individual. So I hope I've conveyed at one level how you can take the same logic and take it to the next level and the next level and the next level. Now, I am not going, I can take this even further. I can take it to the biology of the mitochondria, for instance.
Yeah. Just explain that also, sir, so that it's going to be a different method.
Okay, thank you.
Yeah,
Rajmohan, so I want to just go back, and I hope I've conveyed to you how your concept of Tridosh works at the level of the individual. A person comes to you and you do your pariksha, you do whatever pariksha, rogi pariksha, rog pariksha, and you say, there is a problem in this dhatu, or something is wrong here. And we need to do Shodhana Chikitsa, Shamana Chikitsa, whatever you decide, you are the doctor. Now, the modern biologist doesn't understand these terms, and they say, well, I have a pancreas function. Pancreas is compromised. And you have some other lakshana you use for do the analysis, and you say, this is the problem. Agni is Mandagni here, and I need to push up the Agni.
Yeah. So while we were having the discussion at Yelahanga, we were talking about the cellular energetics and the importance of mitochondria, and we were actually discussing about how our drugs are acting at the cellular level and acting at the mitochondrial level, and in turn of cellular energetics and all those things.
Yeah.
Whether it is possible to demonstrate the activities of some of our rasayanas, etc., in cellular energetics, and what are the methods we can utilize for that, sir?
Okay.
Probably
Thank
after your comments, we can also see comment from Selvan sir. Selvan sir was also commenting on that. We will come to you, sir, soon.
Okay.
Yeah.
Thank you, Rajmohan. So now the question is, can we take it to the next level and further levels? So I want to take you into the world. You asked rightly about mitochondria. You know, mitochondria are the central body of the cell. Let me, for the sake of an image, let me see if I have a page here on, okay, here. Karthik, I'm going to share the screen again, if I may.
Sure, sir.
Okay, here is again, I'll share both these links to you. And this is a very recent article, Rajmohanji, from a magazine called Quanta Magazine, you know. I will link I share chat. It's called Social Mitochondria, Whispering Between Cells Influence the Health. Okay. And mitochondria appear to communicate and cooperate with each other, both within and between the cells. Whenever we say between, that automatically implies communication, some information. Wherever information is moving, it is a shrota. So keep in mind, when we take shrota, we have to look at it at a different level of understanding. You know, shrota, whenever information is moving, it is a path, and that path is a shrota. So whenever we say cells are communicating, mitochondria are communicating, cells, there is a shrota already. This concept of shrotas, if you take it into the world of yoga and Panchakosha theory, then we have sukshma shrotas. There are shrotas which are beyond our senses. What connects Annamaya Kosha to the next level and the Pranamaya Kosha is a subtle shrota. There's a shrota there. And that is represented with the concept of the nadi. So if I come into this, this is what mitochondria would look like within a cell. And then here is Martin Picard, who is presenting his research at the Columbia University. And then he goes on to say that if you look at mitochondria, you will find electromagnetic signals here. Now, it is not a surprise. Wherever there is a movement of a charge particle moves, then you have a signal, electromagnetic signal. A charge moves, there is an electromagnetic signal there. So there is nothing new here about this thing. Now the question, Rajmohanji's question is, can we do some measurement at the level of mitochondria and the DNA to understand Panchabhut Siddhant, Tridosh Siddhant, and to verify Rasayana treatments? Okay. Now, you have an understanding of Rasayanas by saying Rasayanas do certain things. It helps improve the ayana of rasa. It is a Rasayana treatment is something that is done to improve the movement of rasa. So we have taken rasa now. We are going to go into the mitochondria and say, what is the rasa inside the mitochondria? Rasa dhatu, you have used a description, the generic description of rasa dhatu. And I'm saying there are mitochondria there, and rasa has to flow into the mitochondria also to give you perfect Tridosh balance in the mitochondria. So Tridosha is working not only inside the cell, which we showed, but it's also working inside the mitochondria that is inside the cell. This is the scale-independent nature of Tridosha concept. If you look at an aging cell, so Rasayana is intimately connected with the concept of jara, aging. Kalaja or akalaja, whatever it is connected with, and we are saying aging has an impact on Agni and has an impact on Rasayana, will improve whatever is involved in creating a cleaner path for rasa to flow. That is Rasayana. Okay. It could be Rasayana theory, according to Arit Singh and all of our old teachers, is in many forms. Achar Rasayana is also Rasayana. It doesn't have to be just that chemical thing that you have or plant or whatever it is. Achara Rasayana is also an important aspect. It will help prana flow correctly. Okay. Now we'll come to mitochondria. Aging, somebody is aging. One of the common things in aging is what we call the common deletion. Mitochondria has DNA within it. I'm going to put everything in the context of mitochondrial DNA. The total amount of DNA, the size of the DNA molecule in the mitochondria, it's a circular DNA molecule. It is about 17,000 base pairs, 17,000 nucleotides. It's a circular molecule. Okay? Now, as we get older, as we age, as the cell ages, you will find a deletion. About 4,500 base pairs of that mitochondrial DNA is deleted. Now, this 4,500 base pairs of DNA has many genes that code for proteins involved in the whole machinery of ATP production. So when this deletion happens, your cell is compromised for those activities. Now, here is a target that you can measure so easily with the Rasayana treatment. Rasayana is meant to improve Agni. Rasayanas are meant to improve so many other attributes. If I take one cell of any part of the body, and the most accessible might be your blood cells, but be careful when you talk about blood cells, you have so many different types of blood cells. Rakta Dhatu is in fact a collection of cell types. You have lymphoid cells and myeloid cells. Within one of the lineages, you have T cells and B cells. Within that lineage, you have fast cycling cells, neutrophils, which are part of the adaptive, which are part of the innate immune system. They are the ones that attack these viruses as the first step when there is an invasion. They attack bacteria. So we have opportunity to look at one of the many cells in Rakta Dhatu. I'm saying Rasayana treatment, I want to do a DNA analysis. Okay, I will look at Rakta Dhatu. And which of the cells of the Rakta Dhatu? I'll say, look at the neutrophils, because they are rapid cycling cells in the Rakta Dhatu. They are involved in the innate immune system. They have a half-life about 5 days. The cells are cycled every 5 days. If you start a Panchakarma process, by the time you completed your Purva Karma, which might be 5 days to 7 days, you have completely different neutrophils in that person. So even with a simple Purva Karma step, I can measure the quality of mitochondrial DNA in neutrophils just in that first step, Purva Karma step. By the 7th day, when I'm getting ready for the main karmas, the Pradhana Karma, I have 2 time points already in one individual in the neutrophils. I can say, God, this has gone up, this has changed. It is so easy to do. So, Rajmohan, so the point here is Tridosha principles are built on Panchabhuta principles that come out of the Mahaguna principles, which comes out of Avyakt. To go from Avyakt to Vyakt, already you are doing something.
Yeah, so
and from there you're building the next level and the next level and the next level and the next level of complexity, you know. And we have an opportunity to intervene anywhere in there. The challenge for us is to be creative. To just be that creative, to understand this is how we do Purva Karma. This is what I'm going to give this patient for. What are the variations in Purva Karma? Yes, it's a Grita-based procedure. You give a lot of ghee, but for some people, you vary this procedure. You have different types of Grit. I hope I'm correct. You are the clinicians, you know. Yes, you have different types of Grita that you give for different people. And that is matched to the individual's need. Their avastha is matched. So I'm going to stop here. I think Selvan wants to add something, but I will come back to your question, Rajmohanji. I think Selvan has emailed saying he has to go to another meeting. So Selvan, if you wanted to touch on something before you leave, I think the students will like it very much.
Yeah, I think, thank you. And I have to go for a yoga session. Namaste to everybody. Yes. And I had to go for a yoga session, which I committed to online every Sunday. I do it for a group of people. This is just my mother has just informed me about this meeting I joined. Just want to add one element of it is what Mother has beautifully alluded, using a pancreas as an example. In fact, myself worked on the pancreatic cancer and then mucin role and other thing, how the blockage occurs, the ductal role and all. But one other aspect he touched upon is neutrophil. For example, neutrophil is short-lived. It is also promotes inflammation and innate immunity. But what we need to understand in that context is, although the 5 days or 7 days, 5 to 7 days, then by the time you finish your panchakarma, a new emergence of other neutrophils coming into the play. But we have to go one step further is, even some of the short-lived neutrophil can live for longer time in certain situations. And that is what, like for example, in lung fibrosis, and that is exactly happening. And that is the reason it's not just simple equation or the 5 days gone, new emergence of neutrophils coming. Some of them are longer lived, so that we have to understand why the longer living of neutrophil occurs. That creates inflammation. We have to go into the biology of that net neutrophil, either looking at mitochondria or some other aspect, or other enzymes like a DPP1 or other thing. Those are things regulates the net formation, and then that contains inflammation or promoting inflammation. The other scenario, I just want to give a food for thought is, neutrophil, even one hand, one situation, it is a promoter of immunity, and other hand, a promoter of even curtails the growth of cancer cells, I can say, because my own experience in cancer biology. At the same time, in certain situation, it is also one is promoter, other is curtailing the growth of the cancer. It can play a role, it depends on the context. There is a fluidity occurs. We need to understand your explanation from, I don't know much about Ayurvedic principle, but I'm just trying to, I'm a student there, but we have to ask those fluidity, how the fluidity changes. It is not a one simple equation. And that is with that thought only I want to quickly share. And then if you want, I can listen to you for any other comments there, then I would like to go leave the meeting.
Thank you so much, sir.
Thank you, Selvan. Your point is well taken about the pro-inflammatory, anti-inflammatory fluidity in neutrophils. Number two, your point of inflammation in the lung. This is particularly important in the context of this ongoing COVID response.
COVID also, yes, sir.
Problem. So,
yes, yeah.
Many of the formulations you might have used in this ongoing COVID pandemic and the data that you have might uncover some of the issues that Selvan is talking about. We might come to this later on. I don't know whether we will.
Thank you. Thank you.
So there is a question here. Sorry, I'll leave it to you. I hope I've answered your question.
Yeah, yes, sir. Probably I request you to add throw some light into the gut microflora and gut microflora related evaluation, because the gut health, actually, it was a long forgotten aspect. I think now it's gaining much attention worldwide. And of course, we know that how much the Ayurvedic principles and Ayurvedic medicines are caring about the gut health and, of course, the gut microbiome. Sir, please.
Thank you. So, Rajmohan,
and how we can evaluate this panchakarma based on gut microbiome and metabolism aspects.
Thank you. Okay, let me see. I'm going to present it from my a fusion. So I will use terms from both sides, and I think it will convey the meaning I want to convey. Number one is the concept of Mahashrota. So you have a concept of Mahashrota, that the body has within it one large Shrota. So if you look at it in terms of Shrota, the example I always use is how for me, the simplest representation of the human body is like this. You take a paper, you roll it, and you get a Shrota. This is the human body. Okay? So the whole universe and the whole world is running through you, through the Shrota. It's going from one end, from the mouth to the anus, and it engulfs. You are the center of the universe in many ways. This Shrota is the center of the universe, okay? So this is the gut. And within this are organisms. So there are organisms living all the way from the mouth, all the way to the other end of the GI tract. And you, in your text, they're presented as cremies. In a way, it's a little bit mostly correct. They are cremies. That's the term you have used. But when we look at this microflora, you can have bacteria, you can have fungi, you can have viruses also there, different viruses that occur. So if we go from the mouth all the way down to the anus, you will see the oral microflora, first place that you meet microflora, the nasopharyngeal microflora. And there are hundreds of species in each of these locations. Then you go into the stomach, you have certain bacteria that can live at very high pH, and then you go to the small intestine, and then, so these are all, you have terms for all this. You have pakwash, and you have, you have so many grani kind of things, and there are all these things that you understand. Then you go to the large intestine, there is a completely different collection of microbes. And across the section, the population changes. And the most amazing thing is that if your bowel movements are correct on a daily basis or more frequently, then the entire microbial population is refreshed every day. So this is the dynamism. Every day it's cleaning out. If there are problems, if the bowel movement is not, whenever the bowel movement happens, a large part of these microbes are flushed out, and then whatever comes in gets repopulated. Now, this is known in the old text and described for a long time as aspects of Jataragni and aspects of Krimi. Shahaja Krimi, I guess, is what's the term that you people use. Now, when we come to where we are today, this is still a growing area, the area of the microbiome. And as I said, the microbiome is found across the whole body, both inside the Mahashrota and also on the surface of the body and also within the tissue. What happens when you do a karma, panchakarma, for instance? With each of those karmas, you have an opportunity to impact those areas of, impact those microbes in all those different locations. If you do Gandusha and Kavala, for instance, you are impacting the oral microflora. If you're doing Vamana and Virechana, you are impacting the microbes. You have an opportunity to influence the microbes in the upper GI tract. If you're doing Basti, you are doing an intervention that is targeted to the lower GI tract. Now, the procedure for understanding this microbial population and the complexity is getting refined on a daily basis. And the area is called the metagenomics, microbial metagenomics. You look at that entire complexity of microbes. There is a way in which you can prepare the DNA, and you can do the analysis of the microbes that are there, and you can characterize these microbes. Now, one of the most amazing things that came up very early on in microbial DNA analysis and metagenomics is a perfect time dependence of the microbes with age. So when the early observations were done, the scientists said one of the most predictable things that we can do if somebody gave us a fecal sample is to determine the age of the person from where this fecal sample came. This is the tightest correlation that was found very, very early on. There is something there that changes with age. Now, you know how you describe the human body with age, and you know when you start the Panchakarma treatment. So the challenge here is to make those connections, to reach out into the world of microbes, microbiome. The problem here is that amount of data that comes out is the analysis that is involved, the data, the quality of the data is a completely new science in itself. The science of metagenomics, it is computer intensive. It is looking at millions and millions of DNA sequence reads. And so generally you're part of a big institution that's working on this. Here in the UK, they have set up an entire institute for microbiomes in a place called Norwich, which is not far from here. I will send you the link to that. And there are I don't know, there may be about 200 people only researching aspects of the microbiome. This is how sophisticated this is. Now, what I want to add here is that I feel very sad that nobody in this institute has even heard about Panchakarma. Now, I find that sad and disappointing, because here you have a procedure that is described in the oldest textbook of medicine. The most recent redaction was by Agnivesh 2,500 years, 2,500 years ago. This knowledge is much older. But we haven't made a connection with the metagenomic world. The scientists, how much they can learn from our interventions. And so many exciting things will come out. Now, Selvan mentioned about the neutrophils. We can study the impact of a certain treatment on the efficiency of neutrophils, on the energetics of neutrophils. All this is waiting to be done. So I'll give you one other, to put this in the context of the COVID studies. A very interesting study was done in China. Again, I will share the reference. That study looked at the microbiome of people coming to hospitals for acute care, the very serious stages of COVID pathology. And what was amazing is that they all contained a high amount of pro-inflammatory bacteria. So there are certain bacteria that we now associate with inflammation. So here is bacteria that is in the gut that is connected with inflammation. They all have a high incidence. By the time they're present in hospital for acute care, whatever that acute care is, they all, if you look at a sample of the bacteria, very high incidence of pro-inflammatory bacteria. These bacteria are firing up inflammation in that body, and a much reduced amount of anti-inflammatory bacteria. There is a balance of pro-inflammatory, anti-inflammatory bacteria in the gut. That balance is deeply disturbed. How does this happen? We don't know. But I feel there are many clues that we can get from Panchakarma to understand this problem. And more importantly, in the whole area of post-COVID treatment, we are now, UK has now 70 post-COVID clinics, which post-COVID is approximately described, or long COVID as some people call it. Post-COVID or long COVID is a major complication where even after 3 months after they have been relieved of the major COVID symptoms, they are still showing many complications. They feel tired, they don't have energy, they cannot remember things. And it's a complicated problem. And UK right now has about 1.2 million people, 12 lakhs of people with post-COVID problems. And modern medicine doesn't know how to handle this because every individual is different. We need an individualized therapy and management and understanding of this. And this is one of those areas where I feel the complex concepts like Agni, Ampachan, Agni Deepan, all this comes to use. And we must make this dialogue happen. We must see how to make this dialogue happen. So I've touched on two aspects here. In answer to your question about the complexity of the microbes and the microbiome, I've shown you how Panchakarma comes in there and how you can use the different karmas, and you can understand, okay, this is Vamana, it goes on this part, upper GI tract, the stomach, etc. Virechana will take you into the small intestine and cleaning out all of that stuff. And then Basti is specially directed to the lower GI tract, the colonic aspects, and how they interconnect with each other. The neurology for all this is very well understood. As I mentioned before we started, that there is a connection between the colon, the colonic nerves, and back, the muscles of the back, the lower back. If those nerves start firing improperly, then all the muscles of your back are tightening and misfiring, and that gives you backache. For a lot of people, now if you do a simple study, this is just a simple study for any one of your undergraduates, not even for an MD student, for an undergraduate student, maybe final year student, just to do an assessment for all your Panchakarma treatments going on in Government Ayurveda College, Trivandrum, and just ask a question about backache. Patients who come in, do they have stiff back? After Panchakarma, does the backache disappear? I wouldn't be surprised if all your patients said, God, I had a little backache before, that has disappeared after Basti. So there are simple aspects that we already have, but we haven't tuned our data gathering to a point where we can unravel these things, tell the world we have done. I don't know how many Panchakarmas you do every day in Government Ayurveda College, Trivandrum. Every year you might be doing thousands of Panchakarmas.
Yeah, sure, sir. Yeah.
So a simple thing like this, you see, a simple thing like this, we can easily do this interconnection. And in Trivandrum, you are very lucky to be connected with so many institutes. We had people from the interdisciplinary, CSIR interdisciplinary institute. You have Rajiv Gandhi Center. You have infinite opportunities there. It's a well-funded institution. There are people there who are excited about Ayurveda. There is Professor Karta is there, who is working on Amlaki Rasayana and doing many things. It will be easy to excite Rajiv Gandhi Center to interact with our students and have some of our students go and stay in Rajiv Gandhi Center to analyze the gut microbiome, for instance, or look at the mitochondrial integrity in neutrophils in whatever, in patients.
Yeah.
So we have, and Sri Chitra Center, you have another place, which is these are national institutes of excellence, and we have many things that we can do there.
Yeah, thank you, sir. Thank you. There was a very, very thought-provoking comments from your side. I request Dr. Prasad to have to share his thoughts regarding this discussion. And while Prasad are doing that, Karthik, please sort some of the questions appeared there in the YouTube channel, please. Prasad, please.
ഞാൻ പറയുന്നത് കേൾക്കാമോ?
Yes.
ഞാൻ മദൻജിയുടെ കൺസെപ്റ്റുകൾ വളരെ ശ്രദ്ധിച്ച് നിൽക്കുകയായിരുന്നു. കാരണം എന്താന്ന് വിചാരിച്ചു കഴിഞ്ഞാല്, ഈ എങ്ങനെയാണ് ഈ ആധുനിക ഗവേഷണത്തിന്റെ സാധ്യതകളും ആയുർവേദത്തിന്റെ ഒരു റിച്ച്നെസ്, അതിന്റെ ഫിലോസഫിക്കൽ ആയിട്ടുള്ള റിച്ച്നെസ് ആയാലും ശരി, ക്ലിനിക്കൽ ഓറിയന്റേഷനുമായിട്ട് ബന്ധപ്പെട്ടതാണെങ്കിലും ശരി, ഇത് രണ്ടും കൂടെ എങ്ങനെയാണ് ഒരു കോംപ്ലിമെന്ററി ടു വൺ ആൻഡ് അതർ എന്നുള്ള നിലയിലാവുക എന്നുള്ളതാണ് ഞാൻ വളരെ ശ്രദ്ധിച്ചു നിന്നിരുന്നത്. ഇപ്പോഴത്തെ ഒരു സ്ഥിതിക്ക് എന്താണെന്ന് വെച്ച് കഴിഞ്ഞാൽ, വളരെ ഗംഭീരമായിട്ട് പ്രാക്ടീസ് ചെയ്യുന്ന ചികിത്സകന്മാർ തന്നെ വേണം ഇതുമായി ബന്ധപ്പെട്ട ഏറ്റവും പുതിയ ഭാഷയിലും പുതിയ സങ്കേതത്തിലുമുള്ള കാര്യങ്ങൾ വിശദീകരിക്കാൻ കൂടി എന്നുള്ള വലിയൊരു ബാധ്യത നിലനിൽക്കുന്നുണ്ട്. അതേ സമയത്ത് ഇപ്പോ ആദരണീയനായിട്ടുള്ള മദൻജി പറയുന്നത് പോലുള്ള ഒരു ടീം ഈ വിഷയങ്ങളെ പറ്റി വളരെ ഗംഭീരമായിട്ട് പഠിക്കുന്നു. അതുപോലുള്ള ഒരു ടീം ഒരു നല്ല ഒരു സംവാദം നടക്കുകയും ചെയ്യുന്നു എന്ന് വന്നു കഴിഞ്ഞാൽ നിശ്ചയമായിട്ടും ഈ ചികിത്സകന്മാരുടെ മേലുള്ള ഭാരം വല്ലാതെ കുറയുകയാണ്. എന്ന് മാത്രമല്ല, അവര് യഥാർത്ഥത്തിൽ അവരുടെ ആ ഒരു ട്രേഡിലാണ് അവർക്ക് ഏറ്റവും കൂടുതൽ എക്സ്പെർട്ടീസ് വേണ്ടത്. ചികിത്സകന്മാർ എന്നുള്ള നിലയ്ക്ക് അവര് അഷ്ടസ്ഥാന പരീക്ഷയില് ഗാംഭീര്യം ഉള്ള ആളുകളായിരിക്കണം, ദശവിധ പരീക്ഷയില്, ഇന്ദ്രിയങ്ങളെ കൊണ്ടുള്ള പരീക്ഷയില്, ഇതിലെല്ലാം നാഡീ വിജ്ഞാനത്തിലൊക്കെ അവർക്ക് നല്ല വൈദഗ്ധ്യം വേണം. എന്നിട്ട് അവര് അതിഗംഭീരമായ ചികിത്സകൾ നടത്തി അതിൻ്റെ റിസൾട്ട് ഉണ്ടാക്കിക്കൊണ്ടിരിക്കണം. ഇത് ആധുനിക ലോകത്തിന് മനസ്സിലാകുന്ന രീതിയിൽ വിശദീകരിക്കാൻ സാധിക്കുന്നതാണ്. ഇത് വളരെ കൃത്യമായ ചില കൺസെപ്റ്റുകളുടെ പുറത്ത് വർക്ക് ചെയ്യുന്നതാണ് എന്ന് ലോകത്തിനെ കാണിച്ചു കൊടുക്കാൻ നമ്മുടെ ഇന്ത്യൻ സയന്റിസ്റ്റുകൾ തന്നെ ഇപ്പോൾ മദൻജിയെ പോലുള്ള ആളുകളുടെ നേതൃത്വത്തിൽ ഇതിനെ വളരെ കൃത്യമായ ഒരു ഡേറ്റ ബിൽഡ് ചെയ്യാനും അതിന് നമ്മളുടെ തന്നെ കോളേജുകളിലൊക്കെയുള്ള മാൻ പവറുകൾ ഉപയോഗിക്കാനും കഴിഞ്ഞാൽ യഥാർത്ഥത്തിൽ അതൊരു വലിയ അത്ഭുതം തന്നെ സംഭവിക്കും എന്നുള്ളതാണ്. അതേ സമയത്ത് ഇപ്പോൾ സംഭവിച്ചുകൊണ്ടിരിക്കുന്നത് എന്താന്ന് വെച്ച് കഴിഞ്ഞാൽ വൈദ്യന്മാർ വല്ലാണ്ട് കൺഫ്യൂസ്ഡ് ആയി പോകും. വൈദ്യന്മാർ വളരെ ഒരു തരം പരിഭ്രമിച്ചു പോകുന്ന സ്ഥിതി പോലും ഉണ്ട് എന്നുള്ളതാണ്. കാരണം ഒരു സമയത്ത് വെച്ച് കഴിഞ്ഞിട്ട് അവർക്ക് റിസൾട്ട് ഉണ്ടാക്കുകയാണോ വേണ്ടത്, അതോ ഇതിനെ വിശദീകരിക്കുകയാണോ വേണ്ടത്? വിശദീകരിക്കാൻ വേണ്ടി മറ്റ് സങ്കേതങ്ങൾ പഠിച്ചെടുക്കുന്നതിൻ്റെ ഇടയിൽ നമ്മളുടെ തനതായ മാർഗങ്ങളിലുള്ള വൈദഗ്ധ്യം എങ്ങനെയാണ്ട് മാറിപ്പോവും. ഇങ്ങനെയുള്ള ഒരുപാട് ആശങ്കകൾ വന്നുപെടുന്നുണ്ട്. അതിനെല്ലാം മികച്ച ഒരു പരിഹാരമാണ് യഥാർത്ഥത്തിലെ മദൻജിയുടെ ഈ അപ്രോച്ച്. അപ്പോൾ ഒരു ടീം ബിൽഡിങ്ങിന്റെ, മൾട്ടി ഡിസിപ്ലിനറി ആയിട്ടുള്ള ടീം ബിൽഡിങ്ങിന്റെയും ക്ലിനിക്കൽ സൈഡും ഫിലോസഫിക്കൽ സൈഡും, നമ്മൾ എപ്പോഴും പറയാറുള്ളത് പോലെ ഈ തത്വത്തിന്റെയും ശാസ്ത്രത്തിന്റെയും ഭാഗങ്ങൾ വളരെ ഗംഭീരമായി നടക്കുമ്പോൾ തന്നെ അതിലേക്ക് വ്യവഹാരത്തിനെ വളരെ കൃത്യമായി ചേർത്തുകൊണ്ട് പുതിയൊരു മോഡൽ സൃഷ്ടിക്കാനുള്ള ഒരു വലിയ ഓഫറാണ് യഥാർത്ഥത്തിൽ മദൻജി പറഞ്ഞു വരുന്നത് അല്ലെങ്കിൽ മുന്നോട്ട് വെക്കുന്നത് എന്നാണ് എൻ്റെ ഒരു തോന്നൽ. ഇത് കേൾക്കുന്ന സമയത്ത് ധാരാളം സാധ്യതകളാണ് അതിന്റെ മുൻപിൽ തെളിഞ്ഞു വരുന്നത്. ഇതാണ് എനിക്ക് ചുരുക്കി പറയാനുള്ളത്.
താങ്ക് യു പ്രസാദ്. എനിക്കൊന്ന് മുരളി സാറിന് ഈ വിഷയത്തെ കുറിച്ച് സാറിന്റെ ഒരു അഭിപ്രായം ഇപ്പോൾ ഈ ചർച്ച ഇവിടം വരെ എത്തി നിൽക്കുന്ന സന്ദർഭത്തിൽ ഒന്ന് പറഞ്ഞാൽ നന്നായിരിക്കുമെന്ന് എനിക്ക് തോന്നുന്നു.
I am putting my opinions in the chat.
അതെ
രാജ്മോഹൻ, ഞാൻ ചാറ്റിൽ ഇട്ടുകൊണ്ടാണ് ഇരിക്കുന്നത് എപ്പോഴും.
ആണ് സാർ എന്ത് തോന്നുന്നു? സാർ ഒന്ന് പറഞ്ഞാൽ നന്നായിരിക്കുമെന്ന് എനിക്ക് തോന്നുന്നു. If you don't mind.
തീർച്ചയായിട്ടും ഈ കേൾക്കുന്നുണ്ടോ? തീർച്ചയായിട്ടും ഈ തരത്തിലുള്ള സമീപനങ്ങൾ രണ്ട് കാലത്ത്, ദേശകാലങ്ങളെ പറ്റി മദൻജി തുടക്കം തന്നെ നമ്മളോട് അനൗപചാരികമായി സംസാരിച്ചതാണല്ലോ. രണ്ട് ദേശകാലങ്ങളിലുണ്ടായ അന്വേഷണങ്ങളാണ് ഈ ആധുനികവും ശാസ്ത്രവും ആയുർവേദവും ഒക്കെ ആയിട്ടുള്ള ശാസ്ത്രങ്ങളുടെ സമീപനങ്ങൾ. അപ്പൊ ആ ദേശകാലങ്ങളെ കൂട്ടിയിണക്കുക കൂടിയാണ് മദൻജി ചെയ്തതെന്നാണ് എനിക്ക് തോന്നുന്നത്. അത് ഇരു ശാസ്ത്രങ്ങൾക്കും ഗുണം ചെയ്യുന്ന രീതിയിൽ പരസ്പര പൂരകമായിട്ട് തീരും എന്നും കൂടി ഞാൻ വിചാരിക്കുന്നു. അത്രയേ ഉള്ളൂ ഇപ്പൊ എനിക്ക് പറയുന്നത്.
താങ്ക് യു സാർ, താങ്ക് യു. എനിക്ക് തോന്നുന്നത് സംശയങ്ങൾ എന്തെങ്കിലും ഉണ്ടെങ്കിൽ, ഈ ഒരു സമയത്ത് ചോദ്യങ്ങൾ എന്തെങ്കിലും ചോദിക്കാനുണ്ടെങ്കിൽ തീർച്ചയായിട്ടും ചോദിക്കാം. എനിക്ക് തോന്നുന്നു തിരുവനന്തപുരം ഗവൺമെന്റ് ആയുർവേദ കോളേജിലെ അധ്യാപിക കൂടിയായിരിക്കുന്ന ഇന്ദുലേഖ ടീച്ചർ എന്തോ ഒരു ക്വസ്റ്റ്യൻ ചോദിക്കുകയുണ്ടായി. ടീച്ചർ, പ്ലീസ് അൺമ്യൂട്ട് യുവർസെൽഫ് ആൻഡ് ജോയിൻ ദ ഡിസ്കഷൻ ആൻഡ് ആസ്ക് യുവർ ഡൗട്ട്സ് ആൻഡ് എക്സ്പ്രസ് യുവർ കമന്റ് സ്ട്രൈറ്റ് അവേ പ്ലീസ്. യു ആർ വെൽക്കം. ഇന്ദുലേഖ ടീച്ചർ.
Sir, am I audible?
Yeah, yes, please.
Indulekha,
thank you for joining us.
Please continue in Malayalam. I don't know if that is all right. If you feel more comfortable in Malayalam, സംസാരിച്ചും
Sir, I asked one question. Whether the gut microbiome changes, it happens even with an antibiotic therapy. What is the clinical implications? Even with a dietary change, or even with if you drink the different types of water, your flora will change. But what is its clinical implications? Any studies they done so far? I know some studies in AIMS and all doing in Alzheimer's and gut flora, and so many scientists working on that, like Germany, their colonoscopy and gut flora, so many works done. But many of our drugs work like that. Our Virajana, we change our flora, and even an antibiotic can change. Even our Kashan can change flora. And how will we assess that in short time?
Thank you. Your question is very important. Indulekha, you asked a question about antibiotics. I will touch on that initially. We have a lot of studies now on influence of particularly antibiotics on gut microflora. There are a lot of publications. I can send you a list of those publications. If we look at a simple resource, PubMed resource, it is very easy to get those publications also. So you will be able to do that, but I can send you those. It is not only antibiotics. Paracetamol, aspirin, adilor impact. So it does not only that, so there is impact that also. People are taking so many complicated formulations now, some beta blockers or whatever. If you make a long list, everything is impacting that microflora. Adile, how the second effect, the second order effect of that impact on the individual will depend on the individual. Adana Rajmohanji Adyan shows a question. There are many enzymes in the human body and in the gut microflora that will neutralize many aspects of these drugs. You know, if you take things orally, by the time it reaches the gut microflora, there are many things that are going on. From there, it's absorbed and then goes to the liver. There are many enzymes in the liver that will impact the nature of those drugs. And there are studies that have been done in India on assigning some Prakriti aspects with some of the genes, some of the cytochrome genes, etc. That is all published. That is part of the story. But the point here is anything that you take in, we now have tools to see the impact on a daily basis. നമുക്ക് ഇന്നൊരു സാമ്പിൾ എടുത്താല് നമ്മളിന്ന് ആഹാരം കഴിച്ചു, അടുത്ത ആഴ്ച നമ്മൾ ഡയറ്റ് ചേഞ്ച് ചെയ്തിട്ട് ഒരാഴ്ചയിൽ നമ്മൾ ഡയറ്റ് ചേഞ്ച് ചെയ്താൽ we can monitor the change of the microflora. So, and the final point is when we take our kashayams or arishtams, asavams or whatever we are taking, certainly there is an effect on the microflora. And that we need to document. I think we need to make a study at the individual level. Initially, it might be a crude study. We will say we have started, our hypothesis is to study the effect of some aristam kashayam on 1 or 2 microbes. We will say something pro-inflammatory or antibacteria, anti-inflammatory antibacteria, and a common E. coli. And then we can do a simple study to start with. We don't need that sophisticated metagenomic study, etc. We don't need millions and millions of bacteria to be understood. So your question is very, very important and very valid. And I feel that you people in Trivandrum, Government Ayurveda College Trivandrum, is in a very strong position because you get patients. You are doing these treatments every day. All we need to do is put one interface. നമുക്ക് ആ DNA എങ്ങനെയാണ് ഉണ്ടാക്കുന്നത്, fecal bacteria DNA എങ്ങനെയാണ് ഉണ്ടാക്കുന്നത്, അത് പഠിക്കണം. പിന്നെ അത് കഴിഞ്ഞിട്ട് ആരാണ് ഈ അനാലിസിസ് ചെയ്യാൻ പോകുന്നത് നമുക്ക്. അതിനുവേണ്ടി നിങ്ങളുടെ അവിടെ പൂജപ്പരയിലുണ്ടല്ലോ ആളുകൾ രാജീവ് ഗാന്ധി സെന്ററിൽ. അപ്പോൾ നിങ്ങൾ പിന്നെ പറയും ഇതിന് പൈസ എവിടെയെന്നാ വരുന്നത്. അത് we'll have to find the paisa for this. We'll have to write a grant and go to CSIR or Ministry of Ayush or Ministry of Health and Family Welfare and ICMR or something. Now we will have to bring the money. The beautiful thing is all that resources are locally. All we need to do is make the bridges between these two. Prasadji's autism, this area is a very important area. He's saying understanding the Agni, Ojas, Manas as an entity, that is a very beautiful concept. There, our students can work in that project also, because we know that microflora changes are there in autistic children also. Prasadji's treatment is okay. Aharam is a very important intervention. He says Agni is the best way to reach the Manas. Agni will impact Ojas and Agni becomes an entry point into the Manas. It's a very sophisticated idea and that is open to us. അപ്പോൾ അതിനു വേണ്ടി നമ്മുടെ സൈഡിൽ നിന്ന് കുറച്ചുകൂടെ ഒരു ഡിസ്കഷൻ നടത്തിയാൽ we can identify some projects. നിങ്ങളുടെ ഫേവറേറ്റ് കഷായമോ അരിഷ്ടമോ ആസവമോ പഞ്ചകർമ്മയോ whatever it is the intervention, അതിൽ നിന്ന് നമുക്ക് അടുത്ത സ്റ്റെപ്പ് DNA എങ്ങനെയാണ് ഉണ്ടാക്കുന്നത്, that even a school child can do DNA preparation. എന്ന് വെച്ചാൽ these days we have a simple kit. അതിന് you don't need to learn anything. ആ കിറ്റ് യൂസ് ചെയ്താൽ അരമണിക്കൂർ കഴിഞ്ഞാൽ you get the DNA of the highest quality. So it is so easy to do. Yes, we need the funding for doing those studies, but that is a different challenge for us. We have to decide on the project, discuss, connect with Rajiv Gandhi Center, find out what is the costing of the project. That goes into the project management side. Pashe, the initiative must come from people like you, from your situation. Now, that will come out of some discussion, and then we can easily make one project or a series of projects for 6 students or something, you know.
Yeah.
To look at many aspects of Panchakarma or whatever intervention we have.
Okay. Thank you, sir.
So I
audience in the side
Induji, I hope I have answered your question.
Yes, sir. Yes,
Induji?
Okay.
Actually, we are doing a project with Rajiv Gandhi.
Yes, we will discuss. We will discuss this. I would, Induji, I have one question for you.
Okay, sir.
ഞാനൊരു ചോദ്യം ചോദിച്ചോട്ടെ. ഇപ്പൊ നിങ്ങൾക്ക് ഇത്ര ഈ നിങ്ങളുടെ കോളേജിലെ ഹോസ്പിറ്റലിൽ നിങ്ങൾക്ക് ഇത്ര വളരെ വർഷമായിട്ട് പ്രാക്ടീസ് ഉണ്ടല്ലോ, and you have a good experience. Well, can you tell me one condition where this logic can be used? Based on your experience, based on the patients you have seen, based on the responses you have seen, well, now, is there one condition and one treatment, or a treatment, which you think is very effective, where we can apply, where we can develop our project?
Sir, already we are doing something in NFLD. That when we submitted the project, they asked whether mere change in NF, that gut biome is not a condition, but we have to set out other endpoints also. Can I make some more points?
Yes, you can. If you say, I can hear you. Yes, please.
Sir, actually, when we thought of analyzing gut flora, what is its clinical implications? That has to be set up first. So that we took that NFLD, so that if the fibrosis of NFLD comes down, that also, if it also changes the flora, that means it has got some implications. That's why we are working on that with Balakrishnan sir and Chandanu Chattopadhyay, RCGB at Infra.
Now, I have a question, Induji. നിങ്ങളിപ്പോൾ ട്രീറ്റ്മെന്റ് നടത്തുമ്പോൾ നിങ്ങളുടെ ഭാഷയിൽ, ആയുർവേദത്തിന്റെ ഭാഷയിൽ ഇതെങ്ങനെയാണ് എക്സ്പ്ലെയിൻ ചെയ്യുന്നത്? നിങ്ങളുടെ കയ്യിൽ ലക്ഷണങ്ങൾ കാണുമല്ലോ ഇത് ട്രീറ്റ്മെന്റ്സ് നടത്തുമ്പോൾ. സോ അപ്പൊ അതെനിക്കൊന്ന് പറഞ്ഞു തരാമോ? ഈ എന്ന് വെച്ചാൽ ആയുർവേദത്തിന്റെ ഭാഷ
പറഞ്ഞാൽ നമ്മളിപ്പോ പിത്ത വൃദ്ധി എന്ന് പറയുന്നത് കണ്ടീഷൻസ്, അല്ലെങ്കിൽ വാതവൃദ്ധി അല്ലെങ്കിൽ പിത്തവൃദ്ധി എന്ന് പറയുന്ന കണ്ടീഷനിൽ പിത്ത ലക്ഷണങ്ങളൊക്കെ നമ്മൾ വിരേചനം, പിത്ത രോഗങ്ങൾക്ക് അല്ലെങ്കിൽ കാമല എന്ന് പറയുന്ന ജോണ്ടിസ് ലൈക്ക് ഡിസോർഡേഴ്സിൽ വി ഗീവ് വിരേചനം, and after that the symptoms of liver disorder will subside. And what is the mechanism behind that we are searching. One of the theories.
Yes.
That one of the theories, gut leaking is causing liver disorders. That's why we are analyzing gut flora, because it is very close to our gut, and we can easily elucidate that findings. Because when patients get LFT levels rise, we give virejana and some kashayas. That will, we think, we definitely gives pitta shamana, and the principle of pitta is virejana, that is enema. Purgations. So purgations will definitely reduce the pitta. That is our concept. And what causing this change we are analyzing.
Okay, so now you have explained very clearly Pitta Shamana Chikitsa, and that is the logic you have, and you are trying to see what is the connection between your treatment and the outcome. Now, now first question for me, when we try to tighten this protocol, will you see 100% response? Will all your patients respond correctly with your treatment? So you have many treatments for different kinds of lakshanas. Will all your patients respond to your treatment? This is the first question. So this is very important. That is the reason, that lays the foundation for inclusion and exclusion criteria. So when we are trying to take this study towards the molecular side, then we have to tighten many parameters. So you are saying we have lakshanas, we see pitta vriddhi, and we have a standard treatment for this, which is virechana. And this is what we see at the end of the treatment. So you are clear about that. My question is, are they non-responders? That's the first and the simplest question. When we design a research study, then we say, are they non-responders here? Adundo, ningal 100 patient nokkiyal, do you have non-responders where you don't see that effect? Adundo?
Yes, sir. When we do some pilot works, we found that some people that drugs doesn't work.
അതെങ്ങനെയാണ് നിങ്ങൾ എക്സ്പ്ലെയിൻ ചെയ്യുന്ന ഈ നോൺ റെസ്പോണ്ടേഴ്സിന്?
Sir, that we only started and we responded it in clinical conditions. So clinically they are not getting well. They have when we analyzed that sample of around 25 patients, we found that their criteria was different. Actually, in a protocol, we are selecting a group of drugs. So we cannot change that drugs, but that drugs, according to our principles itself, it is not suitable because their dietary pattern is actually very different. Their pattern is very different. So that pattern was actually not suited, but as general protocol, this general protocol is not sufficient for all patients. That is another critical point in our study.
This will, yes, thank you for mentioning this. So you see, we have to look at many other aspects of the study, but it is not impossible, you know.
Yes, sir.
Oh, we can, using your knowledge and your way of thinking, and Ayurveda system and the modern biology system, we can find, but we have to put an interface there. So our experimental design has to be created in such a way that it shows where there is variation and how we can accommodate that variation. But that is not impossible. But it will come through this kind of dialogue. When we talk about this and we say, can we refine the study? Can we refine this experiment? It will come through.
Yes, sir. The major problem I found here is that when we select disease-wise, most probably, I think in this particular study also, they have selected NFALD. And that patient selection is not as per the patient characteristics and individual characteristics as per Ayurveda. That's the that's why this when we take a group of drugs and it may not suitable for some patients, some dosha prakriti or some roga prakriti persons, it may not be applicable. I think audience in questions and please unmute yourself and you can ask. Karthik was making some comments. Karthik, please unmute yourself and talk.
I also, Rajmohanji, there is also one question from let me see if I see this here, Ravi Moosji. Can you explain the benefits of pathya in genetic disorders? Raviji, namaskaram. Kekamo? Thank you for your question.
So I heard that I think you did a research on the genes, how it is changing after doing strict pathya for a long time. So I mean, I heard about that. I don't know whether I'm correct or not. So a strict pathya can change your DNAs or even change your genome. So is that true, or have you done any research on that?
I haven't personally done, but thank you for the question, Raviji. Let me just open up this answer to your question at several levels. One is at the evolutionary level. Presentation, I think there is one slide which is to do with the amylase gene. Namade saliva starch, the first enzyme that interacts with the starch is alpha amylase. And that alpha amylase, there is a gene for alpha amylase. Now, if you look at different populations in the world who have different dietary preferences, Japan, Africa, you look at the number of copies of the alpha amylase gene, salivary alpha amylase gene, you will see the Japanese have about 12 to 14 copies of that gene. versus one of the tribes in Africa which have only 3 copies of the gene. And when we look at their diet, you will see over a very long period of time, the diet has influenced the organization of the genes. But this is over a very, very long evolutionary time. Now, the question you have asked is benefits of Pathya on genetic disorders. So that is at a much shorter time scale. We are looking at the duration of the life of one person, or maybe a few generations. Now, the example of the alpha amylase is over many, many, many generations, a few 100,000 years that you see this effect. If we look at it in the context of the life of one person, 60 കൊല്ലം അല്ലെങ്കിൽ 80 കൊല്ലത്തിന്റെ ആ ഡ്യൂറേഷനിൽ നോക്കിയാൽ what effect will it have? Will we have an effect on genetic disorders? At this point, I'm going to define the genetic aspect in 2 pieces. നമുക്കൊരു DNA level ഒരു genetic change ഉണ്ട്. പിന്നെ there is something called an epigenetic change that is sitting on top of the DNA. That is not a permanent change. It is a flexible change. We can look at DNA, Pathya effects and genetic disorders. If we look at the genetic disorders at 2 levels, the true DNA level change and the epigenetic changes. Then we can start to see the problem separate out even more clearly. That you change the Pathya, you change the food and aspects, then automatically you will see the first thing, which is the epigenetic change. നമ്മളിപ്പോൾ ന്യൂട്രോഫിലിന്റെ കാര്യം പറഞ്ഞത്, അവിടെ നമ്മളൊരു 3 മാസം, if we change the diet, you will see definitely, you will see a lot of epigenetic changes in the neutrophils. ആ ഒരു neutrophils എന്ന് പറയുമ്പോൾ they have a beginning in the Majjadhatu. So you can trace back the origin of this neutrophil through the lineage to Majjadhatu, which is the origin of the hematopoietic cells. All the hematopoietic cells have a Majjadhatu origin. So you can start to see a connection between the effect on Majjadhatu, your food impacting something in Majjadhatu, and then having an effect on the epigenetic aspect of the neutrophils. I give neutrophils as an example because we discussed about this. So there is a way in which we can analyze. So if the effect is seen on the neutrophils, then we can expect a similar effect on other tissues also. The reason why we chose neutrophil is because it's easy to sample. നമ്മൾ ബ്ലഡ് എടുത്താൽ അതിൻ്റെ അകത്ത് നിന്ന് we can isolate very easily. നമ്മളിപ്പോൾ liver നോക്കണമെങ്കിൽ it is slightly more complicated. If we want to study epigenetics or liver tissue, then you have to do a biopsy, and the liver is always evolving. It's such a dynamic organ. You will see changes somewhere there. So it is the same logic can be used. പിന്നെ നമ്മളിപ്പോൾ if we want to look at Astidhatu, it will become very complicated. But we know that in Astidhatu also there is a dynamic process going on. The osteoblast and the osteoclast are constantly in dynamic process. If you can sample it, you can see the DNA effect. But Majjadhatu is easy to see. Medha is very easy to see because you can still sample there. And if you go into all the other dhatus, you will be able to see these effects. I don't know if this gives you an approximate idea on how I see the problem of the effect of Pathya on genetic disorders. I will look at genetics at 2 levels. The true DNA change, which can be at the nuclear DNA or the mitochondrial DNA. I already talked about the mitochondrial DNA with the common deletion, the 4,500 base pairs that is deleted in the context of Raj Mohan's question on rasayanas. But I also want to bring the other aspect of epigenetics. Now, epigenetics, there are chemical modification on the DNA. A methyl group, there is an enzyme that will put a methyl group on the DNA, and then the activity of that DNA changes. It will not express itself, or if you remove the methyl group, it might express again. So that is a very plastic change, and it is definitely impacted with Pathya. Even more amazing is the impact of that from the ritus. Namari 6 ritual undallo, So when we go through the Ritu, you can see a change on expression of the genes in the blood cells, in your adipose tissue, in your Medadhatu. You can see all these things there. Well, it is evident, you can measure it, and your question is completely valid and correct. Yes, the answer in brief is yes, we can see these changes.
Yeah, thank you, sir.
Thank
because
you,
we have
for that question.
few more questions out there. I think Srival sir is here. Sir, please unmute and make your comment, and please ask directly your query. Please, Srival sir, welcome. Welcome to the discussion. And Karthik, please sort out the questions appeared in the chat box, and you may ask to Madan sir straight away. Srival sir.
Namaste, Rajmohan ji.
Namaste, namaste, Srival sir. Please.
Srival ji, namaskaram.
Namaskaram. It was, yeah. Well, I think what is expected out of we are in the dark actually, and then you are throwing good light towards how all we can manage to converse with the biomedical or biomedicine. So that's great. And so there may be, as you told, there may be issues coming out when we try to bridge in these both concepts, but that evolves out and then that gets solved over there is what I also believe. So it's very nice that your presentation will be motivating us to think a lot in the various ways we can approach to explain or sort out things. And so I was also interested that now if we are working on marmas, as we had been always talking, then what all areas, whether it should be at the what you call biochemical level only, plus the electrophysiological level, or how we can think about in the genetical concept also. So that I just put the question because that will be interesting for many also, which many of our students. Madanji, I very clearly tell you that many have that at the tip of their tongue, but they're afraid of opening out or speaking out. That's a typical Kerala phenomenon which I see.
But I think, Srivasi, this is natural, but if we keep our discussions going, then slowly they become, they don't feel as strangers,
So that's why I still as a learner, I am asking you the.
Thank you. Number one, I'm so happy you are here with us. So thank you so much for being with us.
Oh, sure.
Number two, you did your MD in Trivandrum. So that is another big connection with this, with so many other people here who are all in Government Ayurveda College Trivandrum. So that is one. And you are very special that you studied, you went to do MD and you did some work on cancer and you used a Siddha formulation in your study. So you have jumped easily across and you are excited about Marma Chikitsa. So you have jumped, you have shown this flexibility and you are a good model for our students that you can actually move between these disciplines. And most excitingly, you have set up the world's first Ayurveda for sports injury clinic in the world, in Kerala, in Thrissur. You have worked and you have made it happen. And for the students' sake, I just want to say, for the students' sake, I want to say that Srivatji and I, we've been interacting for a long time, number one. And we have been discussing recently about prana and chakras and nadis and marmas and how you can have what is called non-pharmacological interventions for health and well-being. Marma and Varma Chikitsa, especially a specialization that is rich in Kerala. We have the Kalari Chikitsa people who follow this. Kerala, Trivandrum has a very famous Kalari where I know many people who have gone and got treated for a lot of complications. So there is a deep science here, and this area called non-pharmacological intervention is starting to grow in importance. If we go in the direction of non-pharmacological interventions, then we see a whole new universe there of all these things about understanding that is more within the yoga space, yoga asanas, mudras, and the effects. And also you have mantra chikitsa that is at the overlap between Ayurveda and with yoga. And from the yoga side, you can go deeper into the very well-defined aspects, the chakras and the 72,000 nadis, and how people like Srivatsji can manipulate, or the people in the Kalri Chikitsa area can manipulate different marmas and get a physiological response. So there is a big area that is waiting to be understood. The last few weeks, we have been discussing informally in a group. I request all of you who are interested to join us. Please give your name to either Srivatsji or to Rajkumarji, so that you can join in our discussions and listen to what we are discussing now. The question is, what kind of research can we do here? Now, the first important thing is to map the phenomena. So phenomenological mapping. If we take Kerala and maybe Southern Tamil Nadu as the area for this understanding, then let us just make a map of this, what is being done. Different calories, where Kalri Chikitsa is going on, where Marma Chikitsa is going on, who are these people who are doing it? When we go to Coimbatore, we see some very old families that have been doing it for many generations. Srivatsji knows these people. So let us map this thing. Selvan, who was here with us today, he has completed one small paper, which I can share with all of you, on Varma Kalai, Varma Chikitsa, as is known in the Tamil tradition, Siddha tradition, and autism. So I will also share this particularly with Prasadji on how manipulation of the marma points can bring about a physiological change. Now, at first, a lot of people will be skeptical. How can you press certain parts of the body and get a physiological effect? So that is the first problem. And then the second thing, how to measure this. And it's always a conflict there. This conflict of measurement is it is an unfortunate conflict, but we have now to rise up to the challenge and help solve this problem. The first aspect is, how do you press different marma points and get a physiological response? To understand this, in our last event, we had a lady who was with us, Joanna Avison. Joanna is here in England, and she has written a wonderful book called Yoga, and a term which is very important to know, fascia. And it says yoga, fascia, anatomy, and movement. About either 500 page book. She has given the logic of how Ida Pingla. So she is discussing in here how you can understand these concepts, how you can understand the different chakras. What is the meaning of this? From the eyes of one term here, the fascia. Now, to understand the fascia, we have to go into embryology. The fascia is one continuum. It has a tissue structure. It is one continuum that connects everything in the body. You may not understand it from the final form that we have today, but if you go back to embryogenesis, you will see the logic of the fascia. You will see the logic of the Ida and the Pingla. And you will see how the Shushmana is formed out of one interaction between the Ida Pingla. Now, this is what is there in terms of how you bring a response. So anytime you touch anywhere in the body, that effect is communicated across the whole body. We have a possibility to understand simple techniques like Udvartana. There is, I don't know if one of our students from Kottakkal is here with us. If you touch any aspect of the skin, the message is communicated to every other part, including bringing about physiological change, short-term, long-term. If you do a massage, you will see epigenetic change straight away. If you do a series of abhyangas, you will see an effect. നമ്മള് ഒരാഴ്ച അഭ്യംഗ ട്രീറ്റ്മെന്റ് ചെയ്ത് കഴിഞ്ഞാൽ ന്യൂട്രോഫിൽസ് എടുത്താൽ അതിൻ്റെ അകത്ത് you will definitely see an effect. And that is scoreable. It is impossible. You will see, because the moment you touch your skin, you are automatically connecting to Majjadhatu. And that is already described in your text. If you look somewhere, there'll be one shloka that says, this is the reason for Abhyanga. When you do Abhyanga, there is already a connection to the Majjadhatu. And if anything, this input is going to the Majjadhatu, definitely it will affect the hematopoietic lineage, the formation of hematopoietic cells. And definitely in as little as 1 week, you will see an effect on the neutrophils. And if you can do an assay, neutrophils are like amoeboid cells, if you can measure the energetics of that amoeboid movement, you will see in 1 week your neutrophils are moving faster. They are more active. And the reason why they are active is because they have more energy. The mitochondria is changed, the energetics has changed, and they are becoming more active. And you use the term, you make a big jump between Abhyanga and Bala. But in between, there are many, many steps that we have to connect. And one of the steps is increasing the Agni of a neutrophil. The Agni was compromised. And now that has been shifted. And because it has more energy, it is moving much faster now. So you can do all these assays. Neutrophil movement assay is a routine assay these days. Selvan does this, he can measure this thing and he can tell us how to measure neutrophil activity. So there are many opportunities. And this is particularly, I talked about simple Abhyanga. It could be Udvartana. You could have the same effect. It could be just a Marma Chikitsa. Although with the Marma Chikitsa, the initial effect might be a neurological effect. So you might see nerve conduction velocity changes or some of those early changes. And if you sustain those changes, you will see changes in the synaptic architecture. You will see changes in the brain stem. You will see changes in blood pressure. You will see all those other physiological changes, which are understood better in the modern biology area, but explained in the Ayurvedic text in a very compact way. And it will not be found in one section. You might have to go to Granny Chikitsa to understand why something else, why Udvartana works, because it will be in one shloka stuck somewhere else. You might have to look at some other sthana to understand why this thing works. So that is the nature of our text, because it is holding the whole body together, that is interacting in its integrity, And so as far as the text is concerned, it doesn't matter where it is put. It's left to the scholar to dig that information and to put it in the right context. So there is research also to be done in understanding the Samhitas. And when we go from Charaka Samhita lineage to Ashtanga Hridaya lineage, it's even more complicated because it makes it so practical in its activity. The philosophy is left to say, okay, we don't want to worry too much about the philosophy. Give us the day-to-day use of this thing. So that is a complication that I think when we discuss these things, the students will understand, all of us will understand how to look at these different sections. Jvara Chikitsa might have something there which is meaningful for Udvartana. I don't know. There will be one shloka in Jvara Chikitsa that will be useful for explaining some other karma. So I feel that dynamism in the text is both an advantage and a disadvantage. And it is left to us to give examples to students to show the connection between Jataragni and something else, Marma, and to connect with the yoga text and to bring something, to connect with the Siddha text and bring something, to connect with the practitioners, most important, because there are certain practices that have stayed in the families, the family lineage. Yesterday, Narayan Nambi was presenting at one place. I don't know whether many of you were there. And in the Nambi tradition, in the Moose tradition, I think Raviji might be able to tell us, they have a special way of making kashayams. the ratio of water to dravya is very different. And that is unique to their tradition, but it works for them. And we need to understand those kind of details. If we go to Rajasthan, I had somebody who came to who studied with a teacher, Gupta ji from Gujarat, from Nadiad. It's Daniela, our Spanish friend. And she studied master's MSc course in Ayurveda. She was taught by Gupta ji in the Gujarat style of doing Panchakarma. And then she came personally to Kerala to do Panchakarma. And she says, God, you do everything differently here. I thought Panchakarma was done in this way. So even within our traditions, we right now in India, if you go to Maharashtra, they have special treatments that we don't understand. So that dialogue is happening. Fortunately, with these kind of platforms, that dialogue is happening. And I think we need to bring students to appreciate these kind of differences. So Srivatsji's comment is very important. Marma Chikitsa, and how will you measure the effect? I think it is easy to do, but we need to tighten those steps. Just like Induji's protocol that she said, we need to tighten the study. And by discussion, we can tighten those studies and we'll be able to understand better. I'm particularly sensitive to this thing because with Jashna, who might be here in Kottakkal, she was looking at a study on Udvartana for diabetic neuropathy. And will Udvartana help with this, with polyneuropathy? And how will we understand this using Ayurvedic descriptors and the modern descriptors, where you can do single neuron studies if necessary. When you do Udvartana, we have so many neurons that are being affected, and we are applying a dravya. How will you unpack this, and how will you understand? Is it the temperature sensitive neurons? Is it the cold sensitive neurons? It's the warm sensitive neurons? Is it the high pressure sensitive neuron? Which one is being affected? And we can analyze all those little things. So unpacking many of our concepts into small pieces, and we can study even Marma Chikitsa at that level. Marma Chikitsa impacting autism, some aspect of autism, we can unpack that. But our challenge is to keep the integrity of the science, but see the details in a new language, the language of modern biology. Your science is so elaborate. Srivatji, you are saying we are all believers in Advaita Vedanta. You know, Advaita Vedanta makes everything connected. The whole universe is connected. So within that, we see small details. And in that small details, we can do new techniques, new technologies. You can put single neuron recording. You can go to Sri Chitra Center and say, teach us single neuron recording. Our students want to learn this. A student wants to learn how to measure this response at this particular vertebral neuron for Basti. And I'm saying I will look at 4 neurons here. You know, and Basti is the treatment. It will impact these neurons at these parts of the vertebral column. And I want to analyze each of this for each patient. It's a very, very valid study. It must be done because it will uncover new physiology. Yeah. There's a lot of physiology still to be uncovered.
It's great, Madanji.
Thank you, sir. Thank you so much, sir.
Yeah.
I think we are
lucky that
so somewhere we need to wind this up very soon. എനിക്ക് തോന്നുന്നു മുരളി സാറിന് എന്തോ ഒരു ക്വസ്റ്റ്യൻ ഉണ്ടായിരുന്നു തോന്നുന്നു. സാർ മുരളി സാർ പ്ലീസ് ഒന്ന് അൺമ്യൂട്ട് ചെയ്ത് ചോദിക്കാമോ? അതുപോലെ കാർത്തിക്, നമുക്ക് വളരെ വേഗം ഒരു 2 ചോദ്യങ്ങൾ. നമുക്കൊരു 8:30 നമ്മുടെ സമയം, 8:15 നമ്മൾ നിർത്താറുള്ളത്. ഒരു 8:35, 8:40 വരെ നമുക്ക് പോകാമെന്ന് തോന്നുന്നു. അതിനു മുമ്പ് നമുക്ക് വൈൻഡ് അപ്പ് ചെയ്യേണ്ടതുണ്ട്. മുരളി സാർ പ്ലീസ് അൺമ്യൂട്ട് യുവർ സെൽഫ് ആൻഡ് ആസ്ക് ദ ക്വസ്റ്റ്യൻ.
അല്ല, ഞാൻ ചാറ്റിൽ ഇങ്ങനെ ഇട്ടോണ്ടേ ഇരിക്കുകയാണ്.
ഓക്കെ ഓക്കെ.
Murli sir, the question, non-respondent question is a very important one. ഇതില് എനിക്ക് ഒരു എക്സാമ്പിൾ തരാനുള്ളത് ഈ ചൈനീസ് അക്യുപങ്ക്ചറിന്റെ ഒരു സ്റ്റഡി. അവിടെ കാലിഫോർണിയയില് യൂണിവേഴ്സിറ്റി ഓഫ് കാലിഫോർണിയ എർവൈൻ ക്യാമ്പസിൽ അവിടെ ഒരു ഇൻസ്റ്റിറ്റ്യൂട്ട് ഉണ്ട്. ഇറ്റ്സ് കോൾഡ് ദ സൂസൻ സാമുവൽ ഇൻസ്റ്റിറ്റ്യൂട്ട് ഫോർ ഇന്റഗ്രേറ്റീവ് മെഡിസിൻ. അവിടെ they have an interaction with Shanghai, with the Chinese school, acupuncture school. I asked the same question, non-responders, for people who get a ischemic heart attack, they sometimes suffer hypertension, the blood pressure goes up. And is there a non-pharmacological way of containing the high blood pressure? And Chinese said we can put 2 needles here, on the median nerve here, and then we can control the blood pressure. So they were invited to California and they showed it works. Pasha, I had this question about non-responders. Do you have non-responders? And the senior Chinese physician on that day, he said, yes, we do have non-responders. And I said, how do you find out these non-responders? Using your technique, your system, Chinese acupuncture system. Lakshana is very simple. Acupuncture needle, they monitor the temperature of this part of the hand. Acupuncture needle, the temperature changes here. The non-responder, the temperature will not change. They have a diagnostic. This system non-responders in the rainbow. If we look closely, there will be some lakshana. If we take a simple, I'll give you a simple example, Rajmohan. Panchakarma Snehana, you're starting. There will be some people who will give you the correct lakshana after 3 days. And you'll say, okay, now they're ready for the main Pradhan Karma. We can start now. There will be some people who will take 8 days. Now, the question for you is, are there people who don't respond? You are expecting a lakshana, but the lakshana is not coming. There will be. There will be. And so this dialogue with the clinician is very important. Karma eduthalum, there will be non-responders. Now, I'm happy that Murliji is asking this question. And if we go to Kottakkal now, clinical nokiale, you will find always some non-responders. And you might be able to explain it based on your Ayurveda language, which we might be able to explain it, why they are not responding. But we must also now be careful to present those non-responders as something else, as possibly a genetic change, possibly an epigenetic change. We must bring that language into our Ayurveda has always been wonderful in absorbing anything you throw at it. Anything you throw at Ayurveda, it will absorb it, because your logic is based on Panchabhut Siddhanta. Your logic is so extensive. It starts from Avyakt. You can contain everything. Anything that is manifest, it is all a case of where you want to position it in this body of knowledge. So I like this question, and I think your seasoned clinician will be able to tell you, here is a non-responder. Here is a person who responds even quicker. And we must find a new language for that, and we must accommodate that in our discussions. So thank you so much, Murliji, for asking this question.
Sir, a question it was something regarding systems biology and network pharmacological approaches and Ayurveda. I think it is, that question was probably from an interdisciplinary scientist. Sir, could you please answer that question? നമുക്ക് ഈ സിസ്റ്റംസ് ബയോളജി നെറ്റ്വർക്ക് ഫാർമക്കോളജി ടൂൾസ് ഒക്കെ നമുക്ക് എത്രത്തോളം ആയുർവേദത്തിൽ യൂസ്ഫുൾ ആണ്, ഓർ അതിനെ കുറിച്ച് ഒന്ന് കമന്റ് ചെയ്താൽ നമുക്ക് ഈ ഡിസ്കഷൻ അവസാനിപ്പിക്കാൻ സമയമായി സർ. ഓക്കെ,
താങ്ക് യു Rajmohan. So
today's discussion only.
Okay,
today's discussion only.
Okay, so now number one is systems, understanding that network biology. Now, the reason why that whole science has come, Rajmohanji, is because the epistemology of that science, the way that science has begun, is based on, when we take that, ask how did that science come that has given rise to DNA and given rise to the systems, is because they did not have that holistic theory that we have.
Yeah.
So that is the first thing to keep in mind. So they have refined, they said, this is what we can see. We have an instrument. And so this instrument will do all this analysis. It will do analysis. We have mass spectroscopy. That is an analysis that will do all the chemicals here. And so we will apply it to a phenomena to understand something. We come from the other side. We are saying our theory is founded on Panchabhut Siddhanta. We understand the universe in its entirety as having only 5 bhutas. Whatever is manifest is assigned to one of the 5 bhutas. And this is the connection between bhutas. And then we build everything up. Now, what we have is a gap between these two. In fact, all of this is contained in this one, on this side. But they don't understand this language. Oh, you can help them connect. You can give them an example. You can say, here is an effect. We don't use dravyas in isolation. For us, a dravya is a concept because it helps something. Agni doesn't exist by itself. The existence of Agni can be understood by the transformation of something to something else. If this transformation is happening, it automatically means there is Agni somewhere here in the middle. So we can understand Agni not in isolation, but Agni as an effect of something being transformed to something else. They will start searching for Agni and we'll say Agni doesn't exist. Agni exists only when something gets transformed to something else. It's just like saying, this is part of that logic by saying, why does, this is a comment we always make, why does winter exist? Winter as a concept exists, we can say, because autumn has to become spring. Something has to move. There is a movement associated. Why do we have the rainy season? Why do we have Karkadaga Masam? Because the Masam before that has to become the next Masam. So we have Karkadagam in between. That is also logically correct. So in the context of our the question that you have raised, we can go into that space. I want to understand if somebody takes Trikatu, what effect will it have on their Rasadhatu? It's a simple question. And how will I understand this? How will I understand whether there is some aspects of the dravyas that are used in Trikatu? Every dravya, if ginger is grown in Assam, it will have different gunas to ginger that is grown in Kerala. Do you agree with me here? Because it will take the guna of that desha also will come into that dravya. And what you will see is an amazing effect on Rasadhatu, because of that factor. So Trikatu is the same. I'm using the 3 katus, but because I have taken one of the dravyas grown in another desha, it will have a slightly different property. Now you can use a chemical analysis and tell me whether there is more gingerol or whether there is more something or other. That is valid. But from my side, from the Ayurveda side, I don't need to know that. I already have accommodated that by saying, where is the ginger coming from? Is this ginger coming from Africa? Then it has a different guna. It has the guna of that desha already in there. I have factored that. Now, if you tell me the gingerol content is different, it's all right. That is part of guna. That is a part of that guna that I'm taking. So that, you see what I mean? Because your system, your knowledge system has ability to accommodate everything in there.
Yes, sir.
So our challenge now is to bring those two systems together by saying, okay, this is how we understand. Don't run away completely into their language, because then we lose this, because we lose our foundations. We have to stay firm with our foundations. Our foundations are the deepest possible, because it's so logical and it's so elegant in its structure, very elegant. It makes so many different things. I like this, the rasa concept, the Shad rasa concept. It is so amazing because each rasa has two bhutas associated with it. And it's like a full circle. When you go to the last, you start with the first and the last, and you come to Tikta Rasa. When you look at the Bhutik concept of Tikta Rasa, it has the heaviest and the lightest. If you come to earlier rasas, then you see a beautiful combination. It's like playing music. You see one note is combined with the next note, etc., etc. And then it loops back. So this is the elegance of your system. If you run away from this, then you are lost completely. They don't learn about the elegance of your system and how you use it to change physiology. And their system is hanging in the middle of something. It's not rooted on anything. It's rooted on what is the knowledge they have, while your knowledge is rooted at the deepest level, at Avyakt. For me, Avyakt is very exciting, because within Avyakt is where I find the reason for manifestation, because all of this follows after manifestation. Within Avyakt, I'm seeing the three Mahagunas constantly working. And that is the beauty, that this elegance that Sankhya brings into this, the elegance that I find so exciting, that nobody can push you anymore, because I'm starting my theory all the way from Avyakt. And there are the Mahagunas. Sattva, Rajas, Tamas are sitting there, and I understand the mathematics of this. And nobody can push me from that foundation. So I'm very stable, and then I can build up all the way up to what Srivatsji asked about nadis and marmas and chakras and how it affects the body. Yes, it helps when you put in the modern biology language. And I'm hoping that all our students can be introduced to that language.
Yeah, so
and
sure.
I don't think they are lacking in intellectual abilities. They will understand all of this in 5 minutes if you tell them. They have it already. They are already, especially at the MD level, they have it already. Otherwise, they wouldn't have got admission here in Government Ayurveda College Trivandrum or in Kottakkal if they didn't have that ability. So it's just a case of opening spaces, but in opening those spaces, not to forget our tattvas, our shastra, and not to confuse it with vyavahar. I don't even want to discuss vyavahar side here. I want to just constantly focus on the tattvas and the shastras, work on those two principles. And from there, we can connect to anything, anywhere.
Yeah. Thank you so much, sir. Thank you.
There are many more questions here. Kalripayat practitioners say that massage, they can cure Nija rogas also. Yes, Murliji, I agree. And Indulekaji has asked about microtubules and kinesiology changes. And several questions. There is one question that came from somebody with a phone call. It's only identified as Realme 3. Planning a study on changes in gut microbiota of autistic children by giving Falmaja as internal medication as antimicrobial activity. I'm going to answer that question. Also, somebody says, how can they reach me? So, Rajmohanji, I'm just going to put my contact number here for those who want to take this. Plus, so that is my number and also my email address.
എനിക്ക് തോന്നുന്നു വളരെ എലാബറേറ്റ് ആയ ഡിസ്കഷൻസ് ആണ് ഇവിടെ നടന്നത്. എനിക്ക് തോന്നുന്നു സാറിന് ഒരുപാട് കാര്യങ്ങൾ നമുക്ക് സാറിൻ്റെ അടുത്ത് നിന്ന് ഇനിയും കിട്ടാനുണ്ട്. ഒത്തിരി ഡിസ്കഷൻസ് ദിസ് മസ്റ്റ് ഗോ ഓൺ ഇൻ ഡിഫറെന്റ് പ്ലാറ്റ്ഫോംസ്. കാര്യം സാറിനെ പോലുള്ളവരിൽ നിന്നുള്ള ആ ഇൻസൈറ്റ്, കാര്യം ആയുർവേദത്തിൽ തന്നെ നിന്നുകൊണ്ട് വേണം നമ്മൾ ഇതെല്ലാം ചെയ്യണം എന്നുള്ള ആ ഒരു പ്രിൻസിപ്പിളാണ് ഇവിടെ ഏറ്റവും ഉള്ളത്. എനിക്കൊന്ന് അൺഫോർച്ചുനേറ്റ്ലി നമ്മൾ സമയം ഒരുപാട് മുന്നോട്ട് വന്നു. സോ സാർ നമുക്ക് കൺക്ലൂഡ് ചെയ്താലോ, എങ്ങനെയാണ്?
Thank you.
Okay. എനിക്ക് അതിന് കൺക്ലൂഡ് ചെയ്യുന്നതിന് മുമ്പ് some quick comments from പ്രസാദേട്ടൻ, please.
വേറെ ഇനിയിപ്പോ പുതിയതായിട്ട് പറയാനില്ല. നമുക്ക് ഈ സെഷനുകളെ കുറിച്ചൊന്ന് കണ്ടിന്യൂ ചെയ്യുന്നതിനെ പറ്റി ആലോചിക്കാം. ഇപ്പൊ മുരളി സാർ അതിനകത്ത് കമന്റ് ഇട്ടിട്ടുണ്ട്. കാരണം ഈ വിഷയവുമായിട്ട് ബന്ധപ്പെട്ട് നമുക്ക് ചെയ്യാവുന്ന മുഴുവൻ നമ്മൾ എടുത്തിട്ടില്ല എന്ന് തന്നെയാണ് തോന്നുന്നത്.
അതെ, തീർച്ചയായിട്ടും.
മദൻജിയുടെ ഒരു സൗകര്യമുള്ള സമയം കൂടെയൊക്കെ നോക്കി മറ്റൊരു സൺഡേയില് നമുക്ക് ഇതുപോലെ ഒരു സെഷനോ
പിൻ പോയിന്റ് ചെയ്തുകൊണ്ടാവാം.
അതെ 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 അതെ.
എനിക്ക് രാജ്മോഹൻ പ്രസാദ്ജി എന്റെ സൈഡിൽ നിന്ന് എന്തെന്ന് വെച്ചാൽ എക്സൈറ്റ്മെന്റ് എവിടെയാണെന്ന് വെച്ചാൽ നിങ്ങളുടെ you have a long heritage. Even Government Ayurveda College Trivandrum is a long heritage. You're almost 100 and odd years old now. Oh, for you, many things are just natural. It just happens. I don't know, Rajmohan, ningade hospital, etra bed hospital, Ayram?
Almost, sir. Because ipo COVID hospital kude aanu rendu COVID hospital function cheyyunnundu ende ullil.
Ipo nammal Poojapara side poya avade undallo ningalku?
Yeah, yes sir, yes sir.
Inpatients undo avade?
Yeah, undu sir. Pinne rendu campus undu inpatients undu.
Inpatients. Appo idu kando, you have a resource there.
Yeah.
Namukku we have to come up with a way. Pinne north illotu poyale we have so many places, you know. Thiruvananthapuram, another government college. We have so many private colleges. If we go to Kannur, we have another government college. So it is a huge, wonderful resource. What we need is somehow to capitalize. Nobody else, I can tell you something, Rajmohanji, there is no other place in the world where you have these resources.
Okay, sir. Yeah, so, thank you, sir. Yeah.
Idumari resources you don't find anywhere else in the world in such a small space. 35 million people, so many 18 colleges, so many, and so many research institutions. If Srivatsji is connecting with Jubilee Mission Hospital, Thrissur. Oh, there is already some interfacing happening, and we need more of this. Korchuda.
Yeah.
So that is my excitement when I share this. And I'm saying nobody else in the world can do this study that you are doing. And the opportunities nobody else can do. There is no other place in the world where Murliji or people in Kottakkal, the experience is so much. There are many things that you can do that nobody, and we can open up new spaces in medical sciences. That is my excitement.
Yeah.
All our MD students must find a place. Our young students must feel excited. So thank you again for having me with you people. And I have enjoyed this discussion very much. Thank you for the questions. There are many unanswered questions. Murliji, namaskaram. Thank you so much for asking these questions. And we will come back again.
എങ്ങനെയാണ് ഇത്ര അറിവുണ്ടാക്കിയതെന്ന് അത്ഭുതം തോന്നുന്നു. You are my teachers. I learn from Rajmohanji. Every time I listen to him, I'm listening. Prasadji, I'm listening. You know, Agni, Ojas, Manasinde, Akaryam. It is so beautiful. We can become global leaders. We can have entire university on this. You know, Prasadji, autism, we can have a whole institute researching. And the reason is very simple. The reason for need of this, Murliji, Eastern Europe, you will find 20 to 25% of births lead to autistic children. Prasadji, the people who are affected, the child may not be affected too much, you know, because that's how they are born. But the whole family and that entire community, it will be such a blessing if we even can help few people based on this understanding. And Prasadji already has that handle, you know, with the beautiful theory behind it. And the question being asked here is all about that aspect, you know, that there are some dimensions we can handle. Even Western medicine, they don't understand Basti. It's not a treatment. They don't understand. They don't understand how to manipulate Jataragni. That language doesn't exist. Can you see the richness of your language? And Prasadji is saying, I'm going to use Jataragni and improve something. Kottakkal, Dinesh undallo. Dinesh also is in that space. You see, so we already have people there. And we have students wanting to do this work. So that is my excitement. And
Kerala, of course, is a very special land, you know, because we like to do things that are different from other parts of India, other parts of the world. And I feel we haven't even touched a fraction of that potential. We have all that sitting there. And let us see how we can do this. So that is my excitement. I have shared everything with you.
Yeah.
So Rajmohanji, namaskaram. Thank you so much.
Namaskaram, sir. ഓക്കെ, നമുക്ക് നിർത്താൻ സമയമായി. ഇന്നത്തെ ചർച്ച ഞാൻ എടുത്തു പറയുകയാണ്. ഇന്നത്തെ ചർച്ച കാര്യം മദൻ സാറിന്റെ ഒരു എക്സൈറ്റ്മെന്റ് ഉണ്ട്. കാര്യം ഞാൻ വർഷങ്ങൾക്ക് മുമ്പ് സാറിൽ കണ്ട ആ ഒരു എക്സൈറ്റ്മെന്റ്, വളരെ ഒരു യങ്സ്റ്റർ ആയിരുന്ന എന്നെ ആയുർവേദത്തിൽ വളരെയധികം മോട്ടിവേറ്റ് ചെയ്യാൻ ഒരു നോൺ ആയുർവേദിക് പേഴ്സൺ ആയിട്ടുള്ള അദ്ദേഹം ഒരു വളരെ റെപ്യൂട്ടഡ് സയന്റിസ്റ്റ് ആണെന്നുള്ള കാര്യത്തിൽ യാതൊരു തർക്കവുമില്ല. അദ്ദേഹത്തിൻ്റെതായ കോൺട്രിബ്യൂഷൻ സയൻസിൽ ഒരുപാട് കിട്ടിയിട്ടുണ്ട്. നമുക്ക് അദ്ദേഹത്തെ പോലെയുള്ള ആൾക്കാരിൽ നിന്ന് പഠിക്കാനുള്ളത് അദ്ദേഹത്തിനെ പോലെയുള്ള ആൾക്കാരുടെ ആ ഒരു ഇൻക്വിസിറ്റീവ്നെസ്, അതുപോലെ തന്നെ ഡെഡിക്കേഷൻ, ഡിറ്റർമിനേഷൻ, അങ്ങനെയുള്ള കാര്യങ്ങൾ നിന്നുകൊണ്ട് നമുക്ക് നമ്മുടെ ശാസ്ത്രത്തെ എങ്ങനെ കോൺട്രിബ്യൂട്ട് ചെയ്യാം. അദ്ദേഹം ഒരുപാട് പ്രാവശ്യം പറയുകയാണ്, യു നെവർ ലീവ് യുവർ പ്രിൻസിപ്പിൾസ് ബിഹൈൻഡ് ബിക്കോസ് ദോസ് പ്രിൻസിപ്പിൾസ് has its completeness, as it is so comprehensive, it is multidimensional, and ആ ഒരു ലൈഫിനെ കൃത്യമായി എക്സ്പ്ലെയിൻ ചെയ്യാൻ കഴിയുന്ന ആ ഒരു പ്രിൻസിപ്പിൾസിൽ നിന്ന് മാറി നിൽക്കരുതാണ്. സർ, താങ്ക് യു സോ മച്ച് ഫോർ ബീയിങ് വിത്ത് അസ്. സാർ, എനിക്ക് തോന്നുന്നത് വളരെ ഓക്വേർഡ് ആയിട്ടുള്ള ഒരു ടൈമിൽ ഇരുന്നുകൊണ്ടാണ് നമ്മളോട് കൺവേ ചെയ്യുന്നത്. താങ്ക് യു സോ മച്ച്. ഇവിടെ വന്നിരിക്കുന്ന എല്ലാ കുട്ടികൾക്കും അത് വളരെ ബെനിഫിറ്റ് ആയിട്ടുണ്ടെന്ന് എനിക്ക് തോന്നുന്നു. ഇവിടെ വന്ന എല്ലാവർക്കും നമുക്ക് ഈ ചർച്ചകൾ തുടരും സാറിന്റെ അനുവാദത്തോടു കൂടി. സാറിന്റെ ഇനിയും ഒരു സമയം കിട്ടുമ്പോൾ നമുക്ക് മുരളി സാർ പറഞ്ഞതുപോലെ സ്പെസിഫിക് ആയിട്ടുള്ളത് ഒരു ജനറൽ അവയർനെസ് ആണ് നമ്മളോട് ഉദ്ദേശിച്ചത്. സാറിന്റെ ലൈഫും സാറിന്റെ വർക്കും സാറിന്റെ ആയുർവേദത്തെ കുറിച്ചുള്ള അണ്ടർസ്റ്റാൻഡിങ്ങും അതുമായി ബന്ധപ്പെട്ട മെത്തഡോളജീസും എങ്ങനെ നമുക്ക് എല്ലാവർക്കും ഒന്ന് മനസ്സിലാക്കുക എന്നൊരു ഉദ്ദേശമായിരുന്നു ഈ ഫസ്റ്റ് സെഷൻ ഉണ്ടായിരുന്നത്. പ്രോബ്ലി വി വിൽ കണ്ടിന്യൂ വിത്ത് ദിസ് ഡിസ്കഷൻ in a more focused way. Thank you so much, sir. And എനിക്ക് തോന്നുന്നത് കാർത്തിക്, you can as a from the എന്താ പറയാ, നമ്മളുടെ ഒരു യൂത്തിന്റെ റെപ്രസെന്റേഷൻ എന്നുള്ള നിലയിൽ കാർത്തികിന്റെ ഈ സെഷനെ കുറിച്ചുള്ള കമന്റ്സ് പറഞ്ഞുകൊണ്ട് നമുക്ക് ഒരു നന്ദി പറഞ്ഞുകൊണ്ട് നമുക്ക് ഈ സെഷൻ അവസാനിപ്പിക്കാം. From my heart, from my side, from my heart, thank you so much, Madanji, and thank you so much everyone gathered here. We will continue this discussion. Over to Karthik. Karthik, please.
Thank you very much, Rajman. Actually, ഈ സെഷൻ നിർത്തണം എന്ന് തോന്നുന്ന കൊണ്ട് നിർത്തുന്നതല്ല, പക്ഷെ നിർത്തേണ്ടി വരുന്നത് കൊണ്ട് നിർത്തുന്ന ഒരു സെഷനാണ്. കാരണം സമയം ഇത്രയുമായി. ഇനിയും ഒരുപാട് ചോദ്യങ്ങളുണ്ട്. എനിക്ക് തോന്നുന്നു സാറിന്റെ പ്രാണ എന്ന കൺസെപ്റ്റിനെ കുറിച്ചുള്ള സാറിന്റെ അണ്ടർസ്റ്റാൻഡിങ് യഥാർത്ഥത്തില്
ഒരു കാര്യം ആഡ് ചെയ്തോട്ടെ, ഒറ്റ കാര്യം. സോറി ടു ഇന്ററപ്റ്റ് യു. കാര്യം നാളെ മൺഡേ ആയി പോയി എന്നുള്ളത് കൊണ്ടാണ് നമ്മൾക്കത് നിർത്തേണ്ടി വരുന്നത്. ഒരൊറ്റ കാരണം കൊണ്ടാണ്. നമ്മള് സമ്മത ഡേ ആയിരുന്നെങ്കിൽ നമുക്ക് പിന്നെയും കണ്ടിന്യൂ ചെയ്യാമായിരുന്നു. കാർത്തിക്, പ്ലീസ് കണ്ടിന്യൂ.
അപ്പോ പ്രാണനെ കുറിച്ചുള്ള സാറ് ഇൻട്രൊഡക്ടറി നോട്ടിൽ പറഞ്ഞ കാര്യങ്ങൾ അത് ഷെയർ ചെയ്യണമെന്ന് ആവശ്യപ്പെട്ടിരുന്നു. അപ്പൊ അതുപോലെ ഒരുപാട് ഐഡിയാസ് അതുപോലെ കമന്റ്സ് എല്ലാം വന്നിരുന്നു. അപ്പോ മാത്രമല്ല അതില് സാർ ഒരുപാട് റിസർച്ച് പോസിബിലിറ്റീസും മുന്നോട്ട് വെച്ചിട്ടുണ്ട്. യഥാർത്ഥത്തിൽ ആയുർവേദത്തില് വാട്ട് എന്നതിനെ കുറിച്ച് ഒരുപാട് ഉത്തരങ്ങൾ നൽകിയിട്ടുണ്ട്. പക്ഷെ ഹൗ എന്നത് ഏറെക്കുറെ അബ്സ്ട്രാക്റ്റഡ് ആണ്. അപ്പൊ അതിനെ എക്സ്പ്ലെയിൻ ചെയ്യാൻ, എക്സ്പ്ലോർ ചെയ്യാൻ നമുക്ക് ഒരുപാട് ലെവൽസ് ഉണ്ട്. ഇപ്പോ ത്രിദോഷങ്ങൾ എങ്ങനെ പ്രവർത്തിക്കുന്നു എന്നുള്ളതിനെ ഇപ്പൊ ഗട്ട് മൈക്രോബയോട്ടയുടെ ലെവലിൽ സാർ എക്സ്പ്ലെയിൻ ചെയ്തു. എപ്പിജനറ്റിക് മോഡിഫിക്കേഷൻസിന്റെ ലെവലിൽ എക്സ്പ്ലെയിൻ ചെയ്തു. ന്യൂറോളജിക്കൽ, മെക്കാനിക്കൽ, ഇതുപോലത്തെ ഒരുപാട് ബേസിസുകളിൽ നിന്നോട് പറഞ്ഞു. അതിന്റെ ഏറ്റവും മൈന്യൂട്ട് ആയിട്ടുള്ള ഒരു ഡൊമൈൻ ആണെന്ന് തോന്നുന്നു ഈ സെല്ലുലാർ ബയോളജി ലെവലിൽ സാർ എന്ന പാൻക്രിയാസിന്റെ ഉദാഹരണത്തിലൂടെ കാണിച്ചു തന്നത്. അപ്പോ അതിലൂടെ നമുക്ക് ആയുർവേദത്തിന്റെ എവിഡൻസ് ജനറേഷൻ, ആയുർവേദത്തിനെ ഒരു ഗ്ലോബൽ ലെവലിൽ എങ്ങനെ നമുക്ക് പ്രസന്റ് ചെയ്യാം എന്നുള്ളതിനെ കുറിച്ച്, അതുപോലെ സാറ് ത്രിദോഷങ്ങളെ ഏതൊക്കെ ലെവലുകളിൽ നമുക്ക് എക്സ്ട്രാപ്പുളേറ്റ് ചെയ്യാം എന്നുള്ളതിനെ കുറിച്ചൊക്കെ വളരെ ഭംഗിയായിട്ട് പറഞ്ഞു. എനിക്ക് തോന്നുന്നു സാറിന്റെ റെഗുലർ എക്സസൈസിൽ കൂടെയാണ് സാറ് അതിനെ ഏതൊരു ലെവലിലും സാറിന് ത്രിദോഷങ്ങളെ കാണാൻ പറ്റുന്നത്. അത് ഏതൊരു ആയുർവേദ വിദ്യാർത്ഥിയും അതൊരു മോഡലായിട്ട് എടുക്കേണ്ടതാണെന്ന് തോന്നുന്നു. കാണുന്ന ഓരോ സെല്ലിലും, ഓരോ എന്തൊരു വസ്തുവിലും ത്രിദോഷങ്ങളെയും പഞ്ചഭൂതങ്ങളെയും കാണാൻ ഉള്ള സാറിന്റെ ആ ഒരു എന്തൂസിയാസം അത് നമ്മൾ തീർച്ചയായിട്ടും പഠിക്കേണ്ടതാണ്. അതുപോലെ പിന്നെ ഗട്ട് മൈക്രോബയോട്ട, അതുപോലെ സാർ സൂചിപ്പിച്ച ഈ ഗട്ട് മൈക്രോബയോട്ടയുടെ തീമിൽ എനിക്ക് തോന്നുന്നു ഈ ഒരു സങ്കല്പം ഇത്രയും ലൈവ് ആകുന്നതിന് മുമ്പ് തന്നെ നമ്മുടെ ഗുരുനാഥനായിട്ടുള്ള തിരുമുൽപ്പാട് സാറ് കുട്ടികളിലെ ചികിത്സിക്കുമ്പോൾ എനിക്ക് തോന്നുന്നു അവരുടെ ഫോർമേറ്റീവ് ഫേസിൽ വളരെ ഇമ്പോർട്ടന്റ് ആണെന്ന് തോന്നുന്നു. അപ്പൊ അവരിൽ ചികിത്സിക്കുമ്പോൾ സാറ് പലപ്പോഴും കറിവേപ്പില ഇട്ട് നെയ്കൾ കാച്ചി കൊടുക്കുന്നതായി അദ്ദേഹത്തിന്റെ ചികിത്സാനുഭവങ്ങൾ വായിച്ചിട്ടുണ്ട്. അപ്പൊ അദ്ദേഹവും ഈ കോഷ്ടം അല്ലെങ്കിൽ ഗട്ട് എന്നുള്ള ഒരു ആശയത്തെ വളരെയധികം പരിഗണിച്ചിരുന്നു എന്നുള്ളതും തോന്നിയിരുന്നു. അതുപോലെ നോൺ റെസ്പോണ്ടേഴ്സ് എനിക്ക് തോന്നുന്നു ഒരേ ക്ലിനിക്കൽ ഫീച്ചറിന് പിന്നിൽ പോലും വളരെ ഡിഫറെന്റ് ആയിട്ടുള്ള ഈവൻ കോൺട്രഡിക്റ്റിംഗ് ആയിട്ടുള്ള പാത്തോളജിക്കൽ പ്രോസസ്സസ് ഉണ്ടാവാം. അപ്പോ അതും ഈ നോൺ റെസ്പോണ്ടേഴ്സിന്റെ ഒരു പ്രോബബിൾ എക്സ്പ്ലനേഷൻ ആണ് എന്ന് തോന്നുന്നു. ഇപ്പൊ മന്ദാഗ്നി എന്ന് പറയുന്ന ഒരേ ഒരു ക്ലിനിക്കൽ ഫീച്ചറിന് തന്നെ പല പല കോസസ് ഉള്ളതായിട്ട് ടെക്സ്റ്റൽ ഡോക്യുമെന്റേഷൻ തന്നെയുണ്ട്. അപ്പൊ അതുപോലത്തെ എക്സ്പ്ലനേഷൻസ് ഉണ്ടെന്ന് തോന്നുന്നു. പിന്നെ നോൺ ഫാർമക്കോളജിക്കൽ ഇന്റർവെൻഷൻസ്, മർമ്മ ചികിത്സ പോലത്തെ ഏരിയകളും സാർ ടച്ച് ചെയ്തിരുന്നു. മൊത്തത്തില് സാറ് റിസർച്ച് ഐഡിയകൾ പറഞ്ഞു. അതിനോടൊപ്പം തന്നെ സാറിന്റെ മെത്തഡോളജിയും കൂടെ നമ്മളെ പഠിപ്പിച്ചു തന്നു. എങ്ങനെ ഓരോ കാര്യങ്ങളെ സമീപിക്കണം എന്നുള്ളതും സാറിന്റെ വാക്കുകളിലൂടെ സാറിന്റെ ആക്ഷൻസിലൂടെ സാറ് കാണിച്ചു തന്നു. ഏതായാലും ഇത്രയും ഇൻഫോർമേറ്റീവ് ആയിട്ടൊരു സെഷൻ നമുക്ക് തന്നതിന്, സാറിന്റെ ഈ തിരക്കുകൾക്കിടയിലും നമുക്ക് സമ്മാനിച്ചതിന് സാറിന് ഒരുപാട് നന്ദി, ഫൗണ്ടേഷന്റെയും പേഴ്സണൽ ബിഹാഫിലും. അതുപോലെ ഇവിടെ ചർച്ചയിലെ ഇൻപുട്ടുകൾ നൽകിയ മുരളി സർ, പ്രസാദ് സർ, രാജ്മോഹൻ സർ, ബാക്കി ഓഡിയൻസ് എല്ലാവർക്കും ഒരിക്കൽ കൂടി നന്ദി പറയുന്നു. അധികം ദീർഘിപ്പിക്കുന്നില്ല. വൺസ് അഗൈൻ താങ്ക് യു ഓൾ. ഇനിയൊരു ദിവസം കാണാം.
താങ്ക് യു.
Thank you.
Take care.
Thank you. Please feel free to use my email and